When we think of the human body, we often imagine organs such as the heart that pumps blood, the lungs that draw in air, or the brain that quietly governs thought. Yet within us there is another organ, one we cannot see with our eyes or hold in our hands. It is not made of flesh or bone, but of countless living cells that are not our own. This organ is the microbiome, the community of trillions of tiny organisms that share our body and shape our lives in ways both subtle and profound. To picture it is to imagine a hidden world, a quiet forest of invisible life forms, working together in patterns that have been evolving for millions of years. The story of this hidden organ is gentle and vast, and to enter it is to step into a landscape that seems both foreign and intimately familiar. The human microbiome is often described in terms of its immensity. If we were to count all of the bacterial cells living within us, the number would rival or even surpass the total number of human cells in our body. They gather in dense colonies, tucked into the folds of our intestines, spread across the surface of our skin, resting in the moist creases of the mouth, and even lining the airways of the lungs. Some are only temporary visitors, while others remain with us for much of our lives. Together, they form a shifting constellation of life, more diverse and complex than any single organ we could name. And yet, rather than being a burden, these countless organisms create a partnership so deep that to lose them entirely would be to lose part of what makes us human. Scientists have come to call the microbiome a hidden organ, not out of poetic fancy, but out of recognition. Like the heart or liver, it performs functions vital to our survival. It extracts nutrients we could never reach on our own, educates the immune system so that it learns friend from foe, and even whispers chemical messages to the brain that can influence mood, appetite, and thought. It does not sit in a single place, but spreads through the body like an invisible thread, binding together digestion, immunity, and metabolism. To speak of the microbiome is to speak of an organ without boundaries, one that blurs the line between self and other, between the human body and the microscopic life that sustains it. Though modern science has given us names and measurements, the existence of this hidden companion is not entirely new. For centuries, people have sensed that unseen forces accompanied the human body. Traditional medicine often spoke of inner balance, of humors or spirits that needed to be kept in harmony. What was once described in metaphor can now be traced in genes, proteins, and chemical signals. Under the lens of a microscope, the invisible world becomes visible, and we realize that the air we breathe, the water we drink, and the food we eat all bring us into constant contact with a universe of microbes. Each encounter adds threads to the great web of the microbiome, sometimes strengthening it, sometimes testing it. One of the most remarkable qualities of this hidden organ is its diversity. Imagine walking through a dense rainforest, where each patch of soil harbors a different assembly of plants and insects. The microbiome is much the same, a rainforest in miniature, where thousands of species of bacteria, fungi, and viruses coexist in a delicate balance. Some species specialize in breaking down fibers from plants, others in synthesizing vitamins, still others in keeping harmful invaders at bay. This diversity is not random but patterned, shaped by millions of years of coevolution. Just as flowers depend on bees for pollination, we depend on microbes for digestion, defense, and balance. It is a partnership so ancient that neither partner can fully live without the other. In quiet moments, it can be humbling to reflect that much of what we call ourselves is built upon this shared existence. Our cells provide shelter, warmth, and nutrients, while microbial cells provide metabolic power and protection. Together, they form a kind of biological symphony in which each instrument plays a part, often unnoticed, but essential to the harmony of the whole. The microbiome is not something foreign layered onto the body, but a living extension of it, inseparable from our physiology. To speak of you and your microbes is to draw a line that nature itself does not recognize. We are, in truth, a composite being. The study of the microbiome is still young, yet even in its infancy, it has begun to reshape our understanding of health and disease. Where once illness was seen only as a battle between the body and invading germs, we now see that imbalance within the microbial community can tip the scales towards sickness or wellness. A diverse, stable microbiome tends to protect its host, while a disrupted one, stripped of diversity, can leave the body vulnerable. This gentle balancing act is not dramatic, not easily noticed in daily life, yet it underlies so much of what allows us to thrive. To listen to the microbiome is to hear whispers rather than shouts. Unlike a heartbeat, it does not pulse audibly in the chest. Unlike breathing, it does not rise and fall with each moment of air. Its work is quieter, slower, stretched out over hours and days. A fiber of food broken down in the gut becomes a nutrient in the blood. A chemical produced by bacteria drifts through the circulation, reaching a nerve ending and altering its signal. An immune cell, guided by microbial cues, learns patience instead of aggression. These are not sudden events, but gentle ripples across the waters of the body. As we drift deeper into this subject, it is useful to let go of the idea of microbes as enemies. For much of history, bacteria were seen primarily as threats, associated with infection and disease. That view is not entirely wrong. Some bacteria do cause harm, but it is incomplete. In truth, most of the microbes within us are neutral or beneficial, performing tasks that we could not do ourselves. To see them only as foes is like seeing a forest only as a tangle of thorns, forgetting the shelter, the shade, and the nourishment it provides. The microbiome is not a battlefield, but a community. A quiet village of microscopic inhabitants who have learned, over time, to live in harmony with their host. This harmony is delicate, and yet it is also resilient. When we travel, when we eat new foods, when we encounter new environments, the microbiome shifts and adapts. Some species grow more abundant, others retreat, but the overall pattern remains, like a woven fabric that can bend without breaking. It is this resilience that allows the microbiome to endure through the many changes of a human life. Birth, childhood, adulthood, aging. At each stage, the community reshapes itself, reflecting both the constancy of our biology and the variability of our experiences. So as we begin this journey into the microbiome, it may help to hold in mind the image of a hidden organ that is both ancient and ever-changing. It does not beat like the heart or breathe like the lungs, but it hums quietly beneath the surface of our awareness, linking us to the unseen world of microbes that have always been part of life. In the next part of our exploration, we will look more closely at what the microbiome truly is, how scientists define it, and why its vast diversity has earned it a place alongside the organs we already know. But for now, it is enough to rest with the thought that within each of us lies a universe, silent yet vital, a hidden companion carrying out its patient work as we sleep, as we dream, and as we live. 2. What is the microbiome? To understand the microbiome, it helps to begin with the simplest idea. It is the collective name for the trillions of microorganisms that live in and on the human body. These include bacteria, viruses, fungi, archaea, and even tiny, single-celled eukaryotes that do not quite fit neatly into any category. Taken one by one, each of these creatures is microscopic, so small that millions of them can rest on the head of a pin. Taken together, they form a vast and complex community, one that rivals the human body in size and function. The term microbiome does not refer only to the organisms themselves, but also to the genetic material they carry and the ecosystem they form within us. When scientists speak of studying the microbiome, they are studying a living, breathing environment that coexists within our bodies, adapting, responding, and shifting with every change in our lives. The word itself, microbiome, is a relatively recent addition to scientific language, first gaining use in the late 20th century, as new tools allowed scientists to peer into this invisible world. For much of history, microbes were known mostly as the agents of disease. The discovery of bacteria in the 17th century by Antony van Leeuwenhoek, using one of the earliest microscopes, was followed centuries later by the work of Louis Pasteur and Robert Koch, who showed that invisible germs could cause infections. This perspective led to an understandable but incomplete view of microbes as enemies to be eradicated. It was not until researchers began to explore the entire collection of microbes present in healthy individuals that the larger story began to emerge. The microbiome is not just about pathogens. It is about symbiosis, cooperation, and coexistence. In terms of sheer numbers, the microbiome is astonishing. Estimates suggest that for every human cell in the body, there may be an equal or even greater number of microbial cells. The human genome contains about 20,000 genes, but the collective genome of the microbiome, sometimes called the second genome, is measured in millions. This means that much of the metabolic and chemical diversity within us comes not from human DNA, but from microbial DNA. The microbiome, in this sense, expands what it means to be human, endowing us with capacities far beyond those encoded by our own chromosomes. It is as though each of us carries not one blueprint for life, but many, stacked together in a quiet collaboration. The location of these microbes tells us much about their function. The densest population resides in the gastrointestinal tract, especially in the large intestine, where bacteria feed on the remnants of food that our own digestive enzymes cannot fully break down. These bacteria extract energy from fibers, release vitamins, and produce small molecules that nourish the lining of the gut. The skin is another vast landscape where different microbes thrive depending on whether the surface is oily, moist, or dry. The mouth harbors complex communities of bacteria that live on the tongue, the teeth, and the inner cheeks, creating a balance that protects against harmful invaders. The respiratory system, though less densely populated, is home to microbes that influence how we respond to allergens and pathogens. Even the eyes and ears contain their own modest microbial residence. Each site on the body is like a unique habitat, supporting species adapted to that environment, just as deserts, forests, and oceans on Earth support different plants and animals. What makes the microbiome so extraordinary is not just the number of organisms, but the way they interact with us and with one another. These interactions can be cooperative, competitive, or neutral, but together they weave a network of relationships that sustains both the microbes and the human host. A bacterium in the gut may consume dietary fiber and release short-chain fatty acids that nourish intestinal cells. Another bacterium may produce vitamins such as vitamin K or B12, which the body then absorbs. Still another may compete with harmful bacteria for space and resources, preventing them from taking hold. These are not isolated events, but continuous exchanges, like the ebb and flow of a tide, shaping the health of the ecosystem as a whole. The microbiome is also shaped by time. From the moment of birth, the body begins to acquire microbes from the surrounding world. Babies born through the birth canal are coated with microbes from their mother, while those born by cesarean section encounter a different initial community. Breast milk delivers not only nutrients but also beneficial bacteria and sugars that specifically feed certain species in the infant gut. As children grow and encounter new foods, new environments, and new people, their microbiomes diversify and mature. By adulthood, the microbiome reaches a relatively stable state, though it continues to shift in response to diet, travel, illness, and lifestyle. In old age, diversity may decline, often accompanied by changes in health. In this way, The microbiome is not static but dynamic, following the rhythm of life itself. An important feature of the microbiome is its individuality. While certain core groups of bacteria are common to all humans, the precise balance of species varies widely. This individuality reflects diet, geography, genetics, environment, and even chance encounters. Just as no two fingerprints are identical, no two microbiomes are identical. This uniqueness has led some researchers to describe the microbiome as a kind of microbial fingerprint, one that carries a record of where we have been, what we have eaten, and how we have lived. It is a living signature, imprinted not on paper but within the folds of our intestines and across the surface of our skin. Despite this individuality, there are patterns that repeat across populations. For example, people who consume diets high in plant fiber tend to have a greater abundance of bacteria specialized in breaking down complex carbohydrates. Those who eat diets rich in animal protein and fat may host different microbial communities. Geography also plays a role, with traditional, rural communities often showing greater microbial diversity than urban ones. These differences highlight the adaptability of the microbiome, its ability to reorganize itself according to the resources available. In this sense, the microbiome resembles an ecosystem that responds to its environment, flourishing when conditions are diverse and stable, and shrinking when they become narrow or disrupted. The microbiome can be thought of as an interface, mediating the exchange between the body and the outside world. Every meal introduces new molecules into the gut, where microbes help decide how they will be used. Every touch of the skin encounters bacteria, fungi, and viruses, some of which remain, others of which pass through. Every breath carries microorganisms from the air into the respiratory tract. The microbiome receives these inputs, processes them, and returns outputs in the form of nutrients, signals, and defenses. Without this invisible layer, the boundary between the human body and the external environment would be far less resilient. The microbiome acts as a cushion, absorbing shocks, softening exposures, and weaving outside influences into the fabric of the body in a measured way. In describing what the microbiome is, it is tempting to focus on its functions, its diversity, and its importance. But there is also a quieter perspective, the microbiome as a reminder of connectedness. To know that trillions of microorganisms live within us is to realize that we are never alone, that our very survival depends on a web of life far smaller than we can see. It is to remember that life does not exist in isolation, but always in relationship, whether at the scale of forests and oceans, or at the scale of a single human body. The microbiome is, in this sense, a teacher as much as an organ. It teaches us that cooperation is as vital as competition, that balance is as important as strength, and that even the smallest forms of life can shape the course of the largest. So, when we ask, what is the microbiome? The answer is not just scientific but philosophical. It is the hidden organ, made of countless invisible beings. It is the second genome that broadens our abilities beyond what human DNA alone could provide. It is the inner forest that shifts with each stage of life. It is the microbial fingerprint that makes each of us unique. And it is the reminder that health is not a solitary achievement but a partnership, one that reaches down into the very smallest forms of life and extends outward into the whole of the natural world. To understand the microbiome is to understand something fundamental about life itself, that we are never truly separate from the living environment around us, but are woven into it at every level, from the scale of ecosystems to the scale of cells. 3. Why call it a hidden organ? When scientists first began to describe the microbiome as an organ, it was not intended as a metaphor but as an acknowledgement of its profound role in human physiology. Traditionally, the word organ brings to mind a discrete structure that can be seen, touched, and named. The heart, with its muscular walls, the liver, with its smooth lobes, or the lungs, with their delicate branching airways. The microbiome, by contrast, cannot be lifted from the body or placed in a diagram with neat borders. It has no single shape or location, existing instead as a diffuse community spread across multiple regions of the body. And yet, despite this unconventional form, it fulfills so many criteria of an organ that to deny it this status would be to underestimate its importance. To call it a hidden organ is to grant it a rightful place alongside the more familiar structures that sustain life. The first reason the microbiome is considered an organ lies in its essential functions. An organ is defined not only by structure but by what it does. The microbiome performs vital tasks in digestion, metabolism, immune regulation, and communication with the nervous system. It produces vitamins that the body cannot synthesize on its own, such as vitamin K and certain B vitamins. It breaks down complex fibers into short-chain fatty acids that nourish the cells lining the gut. It teaches immune cells the difference between friend and foe, preventing the body from attacking itself while still defending against harmful invaders. These are not minor contributions, but foundational processes without which the human body would struggle to survive. Another reason for the comparison is the microbiome's integration with other organ systems. No organ works in isolation. The heart requires lungs to supply oxygen, the liver processes nutrients absorbed by the intestines, and the kidneys filter the blood delivered by the cardiovascular system. In the same way, the microbiome is woven into the fabric of bodily function. It collaborates with the digestive system to break down food, with the immune system to maintain balance, and with the endocrine system to influence hormones. Signals produced by microbes travel through the bloodstream, affecting distant organs such as the brain and the liver. In this sense, the microbiome is less an isolated unit and more a coordinating hub, a conductor in the symphony of physiological processes. The term hidden organ also reflects its invisibility. Unlike the skin, which forms a visible surface, or the muscles, which move under our command, the microbiome operates quietly in the background. It does not announce itself with beats or breaths. Instead, it works continuously, silently adjusting metabolism, guiding immune responses, and producing chemical signals that drift through the body. Its presence is hidden not only because it is microscopic, but because its contributions are so seamlessly integrated into our daily lives. We do not notice the vitamins it provides, the fibers it digests, or the subtle mood shifts it influences. Only when something goes wrong, when the microbial balance is disturbed, do we begin to see how much we rely upon it. The scale of the microbiome further supports its designation as an organ. With trillions of microbial cells and millions of genes, it is not a minor accessory, but a massive biological system in its own right. The weight of all the microbes in the human body is estimated at 2 to 3 pounds, roughly the same as the brain. If it were possible to gather all the microbes together, they would form an organ in both mass and complexity, comparable to the more familiar organs we learn about in anatomy. This physical presence, though hidden, is a reminder that the microbiome is not just a concept but a material reality. The microbiome also exhibits a remarkable property, often associated with organs: homeostasis. Just as the kidneys regulate fluid balance or the pancreas maintains blood sugar, the microbiome helps sustain internal equilibrium. It adapts to changes in diet, environment, and health status, seeking a stable state that supports the host. This adaptability reflects not chaos but resilience. A healthy microbiome is able to recover from disturbances such as antibiotic use or illness, re-establishing balance through the interplay of its many species. This self-regulating capacity is one of the hallmarks of an organ system, demonstrating that the microbiome is more than a collection of individual microbes. It is a coordinated community that behaves as a whole. There is also a developmental perspective. Organs often have critical periods of growth and maturation, and so does the microbiome. In early life, microbial colonization is essential for the proper training of the immune system. Infants raised in overly sterile environments or deprived of microbial exposure sometimes experience higher risks of allergies and autoimmune conditions later in life. This suggests that the microbiome functions as a developmental organ, guiding the maturation of other systems during key windows of growth, like the thymus, which educates immune cells in childhood and then shrinks with age. The microbiome has a formative role that leaves lasting imprints on the body. The designation of the microbiome as an organ also emphasizes its necessity for survival. Consider how dependent we are on its contributions. Without microbial fermentation of dietary fiber, much of the nutrition in plant-based foods would remain locked away, inaccessible to human enzymes. Without microbial synthesis of vitamins, deficiencies would quickly emerge. Without microbial regulation of immune tolerance, the body would risk constant inflammation. In short, the microbiome fulfills requirements so fundamental that life without it would be dramatically impaired. Indeed, animal studies in which subjects are raised germ-free without any microbiota reveal stunted growth, underdeveloped immune systems, and profound metabolic disturbances. These findings reinforce the idea that the microbiome is not optional but essential, just like the heart or lungs. At the same time, calling it a hidden organ acknowledges the mystery that still surrounds it. Despite rapid advances in sequencing technologies and bioinformatics, much of the microbiome remains uncharted. Many species within the gut have yet to be cultured in laboratories, existing only as genetic fragments in databases. The precise roles of countless microbial genes remain unknown. Even the balance between beneficial and harmful microbes can be difficult to define, as species that are helpful in one context may cause problems in another. The hidden organ is not only hidden because of its invisibility, but because of the vast scientific questions that remain unanswered. To think of the microbiome as an organ is also to shift our sense of self. For centuries, the body has been seen as purely human, composed of our own cells and our own DNA. The recognition of the microbiome challenges this view, showing that we are, in truth, a blend of human and microbial life. Our health depends not solely on our human organs, but on the cooperation of an organ made of non-human cells. This realization softens the boundary between self and other, reminding us that identity is not as sharply defined as we once imagined. We are ecosystems as much as individuals, and the microbiome is the organ that makes this ecosystem thrive. Thus, the name hidden organ carries layers of meaning. It speaks of invisibility, of vital function, of integration with other systems, of resilience and homeostasis, of necessity for survival, and of the mysterious realms still waiting to be uncovered. To give the microbiome this title is to recognize its equal standing with the organs we know more easily. It is to remind ourselves that the body is not just human but more than human, and that life is supported not only by our own cells but by the countless companions that dwell within us. As we continue this exploration, we will see how this hidden organ expands into landscapes as varied as the skin, the lungs, the mouth, and most richly, the gut. Each environment hosts its own community, each with its own patterns of balance and function. But altogether they form one organ, diffuse yet united, hidden yet indispensable. It is a reminder that not all of the body's most important parts can be seen, and that sometimes the greatest treasures are the ones quietly working in the shadows. 4. The landscape of microbes. If one were to imagine the human body as a vast terrain, it would be filled with valleys, rivers, forests, and deserts, each offering its own kind of shelter and resources. The microbiome spreads across this terrain like a quiet population of settlers, finding niches where conditions are favorable and building communities that adapt to local circumstances. Just as the landscape of Earth determines which plants and animals thrive in a given region, The micro-landscapes of the body determine which microorganisms flourish in each environment. The result is a patchwork of ecosystems, some dense with life, others sparse, all contributing to the whole. To explore the microbiome is therefore to explore a hidden geography, one that unfolds not across continents but within the folds of skin, the chambers of the mouth, the winding passage of the gut, and the branching corridors of the lungs. The skin forms the largest of these landscapes, a vast outer layer that serves as both barrier and habitat. Though the skin may appear uniform to the naked eye, to microbes it is a mosaic of microclimates. Some areas are dry, such as the forearms and legs, where only certain hardy species can survive. Others are moist, such as the folds behind the ears or beneath the arms, where microbes that prefer warmth and humidity gather in dense numbers. Still others are oily, such as the forehead or the back, where sebaceous glands provide lipids that particular bacteria consume. Each of these niches fosters its own specialized residents, some of which help protect the skin from invasion by harmful microbes, others that contribute to maintaining its delicate balance. This outer terrain, though constantly exposed to air, water, and touch, is not barren, but quietly alive with microbial activity. Moving inward, the mouth presents a different kind of landscape, one filled with surfaces that are moist, nutrient-rich, and constantly renewed. The tongue, with its textured surface, offers a place for bacteria to anchor themselves, forming communities that can resist being washed away by saliva. The teeth provide hard surfaces where biofilms, or plaques, develop as bacteria cluster together and produce protective layers. The gums and cheeks offer softer tissues where other species find their home. This landscape changes dramatically throughout the day, influenced by what we eat, how often we brush, and even how much we speak or breathe through the mouth. Despite its dynamic nature, the oral microbiome plays an important role in maintaining balance, preventing harmful microbes from taking over while supporting digestion and even influencing systemic health. The lungs were once thought to be sterile, protected from microbial life by the constant flow of air and immune defenses. Yet modern research has revealed that even here, a community exists, subtle but significant. These microbes are fewer in number than those in the gut or mouth, but they still participate in shaping the environment, influencing how the body responds to allergens, infections, and pollutants. The landscape of the lungs is delicate, lined with tiny airways and alveoli, and the microbes that dwell there must withstand constant motion as air rushes in and out. Though less densely populated, the lung microbiome is a reminder that no part of the body is entirely separate from the hidden organ within us. The most extraordinary landscape, however, lies within the gastrointestinal tract. From the moment food enters the mouth to the point where remnants leave the body, it passes through a long, winding corridor populated by countless microbes. The stomach, though acidic and hostile, harbors a few hardy species that can survive its harsh conditions. The small intestine, with its waves of digestive activity, supports smaller populations that aid in nutrient absorption. But it is in the large intestine, or colon, that the microbiome truly comes to life. Here, in the folds and chambers of the gut, trillions of microbes gather, forming one of the densest microbial populations known on Earth. This internal forest hums with metabolic activity, breaking down fibers, releasing short-chain fatty acids, producing vitamins, and generating signals that influence not only the gut itself but organs throughout the body. Beyond these main landscapes, there are smaller but equally intriguing habitats. The reproductive tract, for example, supports specialized microbial communities that maintain protective balance. The eyes, though constantly bathed in tears and exposed to light, still harbor a quiet population of microbes that help guard against infection. Even the ear canal, often thought of only as a passage for sound, provides a home for microbial residents adapted to its unique conditions. Each of these niches demonstrates the adaptability of microbial life, its ability to find a foothold in places where conditions may seem inhospitable to us, but perfectly suited to them. What makes these microbial landscapes particularly fascinating is the way they differ from one another, while still contributing to the unity of the microbiome as a whole. The bacteria that thrive on the skin are not the same as those in the gut. The species that flourish in the mouth may not survive in the lungs, and yet together they form a connected system, each part influencing the others through shared signals, immune responses, and metabolic products. Just as ecosystems across the planet interact through air currents, rivers, and migrations of species, the microbial ecosystems of the body interact through circulation, immunity, and communication across tissues. These landscapes are not static. They change over time and in response to the environment. A shift in diet can alter the composition of the gut microbiome within days. A change in humidity can reshape the microbial balance on the skin. Even emotional stress can influence the microbiome indirectly, as stress hormones affect immune responses and gut activity. The landscapes of the body are therefore more like living gardens than fixed territories, constantly shifting, adapting, and responding to the seasons of life. It is worth pausing to imagine this geography with gentle attention. Picture the skin as a desert plain dotted with microbial oases, the mouth as a moist valley crossed by rivers of saliva, the lungs as branching caves with hidden niches, and the gut as a dense forest teeming with unseen life. Though we cannot walk through these landscapes with our feet, we carry them with us wherever we go. They are not alien territories but intimate parts of ourselves, coexisting with our human cells to sustain health. To call the microbiome a landscape is therefore to recognize its diversity, its adaptability, and its interconnectedness. Each site on the body offers a distinct terrain, each populated by microbial communities that have learned to thrive in their unique environment. Together, these landscapes form a hidden geography within us, one that mirrors the complexity of the natural world outside. By understanding this geography, we begin to see that the human body is not a single entity but a collection of habitats, each alive with life too small to see yet too important to ignore. And so, the landscape of microbes is not merely a scientific curiosity. It is a reminder that within us lies a world as varied as mountains and seas, deserts and forests. It is a world that has grown with us since birth, that changes as we change, and that will continue to shape our health for as long as we live. To honor these landscapes is to honor the hidden organ they compose, and to recognize that even in the quietest corners of the body, life is abundant, intricate, and enduring. 5. The gut microbiome, a vast inner forest. Deep within the winding corridors of the digestive tract lies a world unlike any other in the human body. It is dark, warm, and filled with nutrients that pass by in a steady flow. To most people, this space is simply the gut, a place where food is broken down and prepared for absorption. But when viewed through the lens of microbiology, it becomes something much more, a thriving forest of microbial life, dense and intricate, with layers of interactions that mirror the richness of natural ecosystems. This hidden forest is what scientists call the gut microbiome, and it is by far the largest and most diverse community of microbes within us. The gut microbiome begins its story at birth. As an infant enters the world, their sterile digestive tract is quickly colonized by bacteria from the mother and the surrounding environment. These early settlers pave the way for others, shaping the conditions in which new microbial species can thrive. Breastfeeding adds to this early landscape, as breast milk contains not only nutrients but also specialized sugars designed to nourish beneficial bacteria. By the time a child reaches the end of their first year, their gut is home to a growing and diverse microbial community. Over time, this community becomes more complex, stabilizing into an adult-like form. This early development is not merely incidental. It sets the stage for how the immune system, metabolism, and digestion will function for years to come. The scale of the gut microbiome is immense. It is estimated that trillions of microorganisms live in the gut, representing thousands of species. The colon, in particular, is considered one of the densest microbial habitats on Earth, surpassing even the richness of soil. Within just 1 gram of intestinal content, there may be more microbes than there are humans on the planet. These microbes are not scattered randomly, but organized into communities that interact with one another and with the host. Some species occupy the mucus lining of the gut wall, where they form protective layers. Others thrive in the central contents of the colon, feeding on the remnants of digested food. Together, they form a dynamic ecosystem, constantly shifting in response to what we eat, how we live, and even how we feel. One of the most vital roles of the gut microbiome is in digestion. Human enzymes are capable of breaking down many nutrients, such as proteins and simple carbohydrates, but they cannot digest certain fibers found in plants. These fibers would pass through the digestive system unused if not for the intervention of gut microbes. Bacteria in the colon ferment these fibers, producing short-chain fatty acids such as acetate, propionate, and butyrate. Molecules are then absorbed by the gut and serve as fuel for the cells that line the intestinal wall. In this way, the microbiome transforms otherwise indigestible material into vital energy sources, demonstrating how deeply intertwined our survival is with microbial activity. Beyond energy production, the gut microbiome contributes to the synthesis of vitamins and other compounds that the body cannot produce on its own. Certain bacteria generate vitamin K, which is crucial for blood clotting, while others produce B vitamins, essential for metabolism. These contributions may seem small, but without them, deficiencies would quickly arise. The microbiome also influences the metabolism of bile acids, cholesterol, and even certain drugs, revealing its role not only in nutrition but in pharmacology and overall physiology. The forest-like nature of the gut microbiome is most evident in its diversity. Just as a healthy forest contains many species of trees, plants, and animals that maintain balance, a healthy gut contains a wide variety of microbial species. Some are dominant, making up large portions of the population, while others are rare but still important. This diversity creates stability. If one species declines, others can often take over its role, preventing collapse. When diversity is lost, however, the system becomes fragile, leaving room for harmful bacteria to take hold. This state of imbalance, known as dysbiosis, has been linked to a range of conditions, from digestive disorders to metabolic diseases. It shows that the health of the gut microbiome is not determined by the presence of a single species, but by the overall harmony of the ecosystem. Communication is another key feature of this inner forest. The gut microbiome produces chemical signals that influence not only the gut itself, but also distant organs. Some of these signals travel through the bloodstream to the liver, where they affect metabolism. Others reach the immune system, teaching it tolerance and preventing unnecessary inflammation. Still others interact with the nervous system, forming part of the so-called gut-brain axis. Through this axis, the microbiome can influence mood, stress responses, and even cognitive functions. It is remarkable to consider that tiny bacteria existing in the depths of the colon can send signals that alter how the brain perceives the world. The structure of the gut also supports this microbial community. The lining of the intestine is not smooth but covered in folds and tiny projections called villi and microvilli. These structures increase the surface area for absorption, but they also create niches where bacteria can settle and interact with host cells. The mucus layer that coats the gut provides both protection for the host and food for certain microbes, creating a constant exchange between human tissues and microbial life. This close contact allows for dialogue between species, with the host providing shelter and nutrients, while the microbes offer metabolic and defensive services in return. Diet plays a profound role in shaping this inner forest. A diet rich in plant-based foods, filled with fibers and complex carbohydrates, tends to support a diverse and resilient microbiome. In contrast, diets high in processed foods and low in fiber can reduce diversity and encourage the growth of species associated with inflammation. These changes can occur quickly. Studies have shown that altering the diet can reshape the gut microbiome in just a few days. Yet, the long-term stability of the microbiome depends on consistent nourishment, much like a forest thrives only when its soil and water are maintained. The foods we choose each day act as seeds and sunlight for the microbial communities within us. The gut microbiome also reveals its forest-like character in its response to disturbance. Just as a forest can be damaged by fire, logging, or drought, the gut microbiome can be disrupted by antibiotics, illness, or stress. These disturbances may wipe out entire populations of microbes, leaving behind gaps that take time to heal. In many cases, the microbiome is resilient, regrowing its diversity and returning to balance. In other cases, recovery is incomplete, and the ecosystem remains altered. This resilience and vulnerability mirror the delicate balance of natural ecosystems, reminding us that the health of the gut is as dependent on stability as the health of a forest. It is also striking to realize that this forest is not separate from the outside world but constantly connected to it. Each bite of food introduces new molecules that microbes transform. Each sip of water, each touch of soil, each interaction with the environment adds to the exchange. The gut microbiome is not sealed away but open to the world, adapting continuously to what enters the body. In this way, it serves as a bridge between the external environment and internal physiology, translating the raw material of food and drink into forms the body can use. In reflecting on the gut microbiome as a vast inner forest, one sees not just an organ system, but a microcosm of life itself. It contains diversity, interdependence, communication, resilience, and vulnerability. It grows with us from birth, shapes our health throughout life, and continues to shift as we age. To walk through this forest in imagination is to encounter countless species, each small but essential, forming a living system that sustains us in silence. Though hidden from view, it is as real and as vital as the forests that cover the earth. In caring for it, we care not only for our own health but for the intricate partnership between human life and microbial life that has existed for millennia. 6. Nutrition and digestion. The process of eating is so familiar that it often feels simple. We take in food, chew it, swallow it, and feel satisfied. Yet beneath this everyday act lies a complex and finely tuned system, one that involves not only the body's own organs but also the countless microbes that dwell within the gut. Nutrition and digestion are not tasks carried out by human cells alone. They are shared responsibilities, divided between host and microbiome, in ways that reflect millions of years of partnership. To understand how deeply the microbiome is woven into our nutrition is to see that we are not solitary beings feeding ourselves, but communities, nourished by an intimate cooperation with invisible life. When food enters the mouth, digestion begins with the physical act of chewing and the chemical action of saliva. Enzymes in saliva break down starches into simpler sugars, while the teeth grind food into smaller pieces. At this stage, the microbiome is already at work. The oral cavity contains its own set of microbial communities that interact with food particles, sometimes beginning to ferment carbohydrates before they even reach the stomach. These microbes are not the main players in nutrition, but they hint at the role microbes will take on more fully in the gut, transforming food into forms that the human body can use. As food travels down the esophagus and enters the stomach, it encounters an acidic environment designed to break down proteins and eliminate most harmful microbes. Yet even here, a few microbial species endure, finding ways to survive the acidity. They remind us that the digestive system is never completely empty of life, even in places that seem hostile. But it is only when food moves into the intestines that the microbiome takes on its central role. The small intestine absorbs sugars, amino acids, and fats through the lining of villi and microvilli. Yet much of the fibrous material from plants passes through undigested. This is where the microbes of the large intestine step forward, performing the tasks that human enzymes cannot accomplish. Fibers are the most striking example of this cooperation. Human digestive enzymes cannot break down many of the complex carbohydrates found in fruits, vegetables, grains, and legumes. Without microbes, these fibers would simply pass through the body unused. Instead, bacteria in the colon ferment these fibers, breaking them into smaller molecules, known as short-chain fatty acids. Among these, butyrate, acetate, and propionate stand out for their importance. Butyrate, for instance, is the preferred fuel for the cells that line the colon, helping to maintain the integrity of the gut barrier. Acetate and propionate enter the bloodstream, where they influence metabolism in the liver and even send signals to the brain. In this way, dietary fiber becomes not just roughage but a vital source of energy and communication, thanks to the microbiome's hidden work. Proteins, too, are shaped by microbial action. While the stomach and small intestine break proteins into amino acids, microbes in the large intestine continue the process, fermenting certain amino acids into compounds that affect health. Some of these compounds can be beneficial, while others, in excess, may contribute to inflammation or disease. This duality highlights the delicate balance of microbial metabolism, where outcomes depend on diet, microbial diversity, and overall health. It is a reminder that nutrition is not only about what we eat, but about how microbes interpret what we provide. Fats are less directly influenced by microbes, yet even here the microbiome plays a role. Bile acids, secreted by the liver to aid in fat digestion, are modified by gut bacteria into secondary bile acids. These altered compounds influence not only fat metabolism, but also cholesterol balance, immune function, and even the composition of the microbiome itself. This cyclical relationship, where microbes shape bile acids and bile acids shape microbes, underscores the interconnected nature of nutrition and microbial ecology. Beyond macronutrients, the microbiome contributes to micronutrient synthesis. Certain bacteria produce vitamins that the body cannot make, such as vitamin K, essential for blood clotting, and several B vitamins, crucial for energy metabolism and nervous system function. These microbial vitamins enter the circulation and become part of the body's nutrient supply, demonstrating how microbial activity extends beyond digestion to contribute directly to essential physiological needs. What makes microbial digestion so remarkable is not only its contribution to nutrition, but also its influence on metabolism and appetite. Short-chain fatty acids produced during fermentation do more than provide energy. They act as messengers, binding to receptors on cells and influencing hormone release. Some of these hormones regulate appetite, signaling fullness after a meal. Others affect insulin sensitivity, linking the microbiome to blood sugar control. In this way, microbes help govern not just the processing of food, but the body's overall relationship with hunger, satiety, and energy balance. The cooperation between diet and microbiome is not static but dynamic, shifting with what we consume. A diet rich in diverse plant fibers fosters microbial communities that excel at fermentation and vitamin production, creating an ecosystem that supports resilience and health. A diet low in fiber but high in fat and processed sugars may reduce microbial diversity, encouraging species that thrive on simple carbohydrates but offer fewer benefits. These shifts can happen quickly, sometimes within days, reflecting the microbiome's ability to adapt. Yet long-term dietary patterns leave lasting imprints, shaping the stability and richness of the microbial forest within. One of the most profound aspects of this cooperation is its subtlety. We rarely think of ourselves as relying on bacteria to process our meals, yet every bite of an apple or spoonful of lentils becomes nourishment only because trillions of microbes ferment fibers into usable molecules. We rarely think of microbes influencing our feelings of fullness, yet hormones released in response to microbial products help determine when we stop eating. The microbiome's role is not loud or dramatic but woven quietly into the rhythm of daily life, guiding the body's interaction with food in ways we cannot consciously perceive. This partnership also underscores the idea that nutrition is not a solitary act. Eating does not nourish only human cells but microbial ones as well. Each meal is a feeding not just of ourselves but of our microbial companions. They, in turn, repay us by producing compounds that protect the gut lining, support immunity, and regulate metabolism. This mutualism means that the quality of our food influences not only our own health but the health of our microbial communities. To feed ourselves well is to feed them well, and to feed them well is to ensure they continue to sustain us. In viewing nutrition and digestion through the lens of the microbiome, one begins to see the human body as more than an individual organism. It is an ecosystem where host and microbes collaborate to transform food into energy, signals, and health. This perspective broadens the meaning of nourishment, extending it beyond the absorption of calories and nutrients to include the cultivation of a thriving microbial community. The food we eat is not just sustenance for us. It is the foundation upon which the hidden organ of the microbiome builds its vital contributions. And so, nutrition and digestion become not simple processes but shared journeys. Each meal sets into motion a cascade of human and microbial actions, unfolding silently within the depths of the gut. Each nutrient becomes part of a dialogue between host and microbe, shaping health in ways both immediate and long-term. To eat, then, is to participate in an ancient partnership, one that has sustained human beings for countless generations and will continue to do so for as long as we share our bodies with the invisible forest of life within. 7. The microbiome and immunity. The human body is often described as a fortress, defended by an immune system that guards against invasion. Yet within this fortress lives a teeming population of microbes, numbering in the trillions, existing not as enemies but as allies. To understand this paradox, one must look at the deep partnership between the microbiome and the immune system. Far from being separate, these two entities are intertwined, each shaping and guiding the other in ways that are subtle, enduring, and essential for life. The immune system is not simply a shield raised against all microbes. It is a finely tuned network that distinguishes between friend and foe, between those who support life and those who threaten it. The microbiome plays a central role in this education, teaching the immune system tolerance, guiding its responses, and standing as a first line of defense against harmful invaders. From the earliest moments of life, the microbiome begins to instruct immunity. At birth, a newborn's immune system is untrained, ready to respond but lacking the experience to distinguish danger from harmless presence. The microbes encountered during birth and early feeding provide the first lessons. They expose immune cells to antigens molecules that trigger recognition and help the system learn which signals should prompt defense and which should be accepted as part of the body's environment. This early training is vital. Infants who grow up in overly sterile conditions, deprived of microbial contact, sometimes develop immune systems that overreact to harmless stimuli, leading to allergies or autoimmune conditions later in life. The microbiome, in this sense, is like a tutor, showing the immune system how to balance vigilance with restraint. The relationship between microbiome and immunity is most visible in the gut, where the densest populations of microbes reside. The lining of the intestines is covered in immune cells, forming what is known as the gut-associated lymphoid tissue. This tissue acts as a watchtower, constantly sampling microbial products and deciding how to respond. Beneficial microbes reinforce tolerance, releasing compounds that calm inflammation and encourage regulatory immune cells to develop. Harmful microbes, by contrast, provoke alarm signals that summon defensive responses. The result is a system that can tell the difference between friendly inhabitants and dangerous intruders, a distinction that would be impossible without the steady guidance of the microbiome. One of the most fascinating aspects of this dialogue is the way microbes influence immune signaling. Short-chain fatty acids, produced when bacteria ferment dietary fibers, act as messengers to the immune system. These molecules encourage the growth of regulatory T cells, a class of immune cells that suppress overreactions and maintain tolerance. Without this microbial input, the immune system is more likely to become erratic, attacking harmless antigens or even the body's own tissues. Other microbial products, such as polysaccharides and metabolites, similarly shape immune responses, fine-tuning the balance between defense and tolerance. In this way, the microbiome acts as both mentor and moderator, ensuring that the immune system responds when needed, but does not exhaust itself with unnecessary battles. The microbiome also provides direct defense against pathogens. By occupying space and consuming nutrients, beneficial microbes prevent harmful species from establishing themselves. This phenomenon, often called colonization resistance, is a powerful form of protection. A healthy gut microbiome can resist infection from dangerous bacteria simply by denying them room to grow. In addition, many microbes produce antimicrobial compounds that inhibit or kill competing species. These natural antibiotics are not indiscriminate. They target specific rivals while leaving the broader community intact. This microbial competition mirrors the ecological dynamics of forests and grasslands, where diversity maintains stability and prevents domination by a single invasive species. The skin microbiome offers another example of microbial defense. Bacteria that naturally live on the skin produce substances that lower the pH, creating an environment less hospitable to pathogens. Some species even secrete compounds that directly kill harmful bacteria, acting like quiet guardians patrolling the surface of the body. These protective microbes are supported by the immune system, which learns to tolerate their presence while remaining ready to respond to outsiders. This cooperation illustrates how immunity is not a solitary shield but a collaborative effort, one that relies on microbial partners to maintain the body's barriers. Even the respiratory tract, once thought to be largely sterile, reveals this partnership. Microbes living in the airways influence how the immune system reacts to allergens and infections. Studies suggest that a balanced airway microbiome may reduce the risk of asthma and other inflammatory conditions, again demonstrating how microbial presence can guide immune tolerance. Without these subtle influences, the immune system might treat harmless particles like pollen as dangerous threats, triggering unnecessary inflammation. The microbiome thus helps tune the sensitivity of immune responses, much like adjusting the strings of an instrument until the sound is harmonious. Disruptions to the microbiome can unsettle this balance, leading to immune problems. Antibiotics, though life-saving, can wipe out beneficial microbes along with harmful ones. In the aftermath, the immune system may struggle to recalibrate, sometimes overreacting or leaving gaps that pathogens exploit. Dysbiosis, a state of microbial imbalance, has been linked to conditions such as inflammatory bowel disease, allergies, and autoimmune disorders. These associations highlight the importance of microbial guidance. Without it, the immune system can lose its sense of proportion, either attacking too fiercely or failing to defend effectively. The connection between microbiome and immunity also extends to communication with the brain. The gut-brain immune axis describes how microbial signals can influence not only immune cells, but also mood and stress responses. Stress hormones alter immune function, which in turn can affect microbial balance, creating a cycle of interaction that ties mental health, immunity, and the microbiome together. This interconnectedness suggests that caring for the microbiome may support not only physical defense but also emotional well-being. The immune system is not isolated. It is embedded within a larger web of communication that includes microbes and the nervous system. In reflecting on this partnership, it is striking how the immune system and the microbiome mirror each other. Both are vast, complex, and adaptable. Both are essential for survival, yet neither can function properly without the other. The immune system provides the rules, distinguishing between danger and safety, while the microbiome provides the context, teaching those rules through constant interaction. Together, they create a dynamic equilibrium, one that sustains health quietly and continuously. To view the microbiome as part of the immune system is to soften the boundaries we often imagine between self and other. We tend to think of immunity as defense against outsiders, but much of its strength comes from collaboration with those very outsiders, microbes that have become part of us through long coevolution. They are no longer simply foreign. They are partners, woven into the immune fabric of the body. Their signals shape our defenses, their presence trains our tolerance, and their competition keeps threats at bay. Without them, the immune system would be like a soldier without training, armed but directionless, ready to attack but uncertain when and how. Thus, the microbiome and immunity cannot be separated in any meaningful way. They are parts of the same system, two aspects of a single process that keeps the body safe, balanced, and resilient. To recognize this is to see that defense is not about exclusion but about harmony, about creating a stable environment where both human and microbial life can thrive. The microbiome is not only a hidden organ of digestion and metabolism, but also a hidden organ of immunity, standing silently between us and the countless microbial worlds beyond. Its lessons are simple yet profound. Tolerance is as important as vigilance, balance as vital as strength, and cooperation as enduring as competition. Communication with the brain. Among the many surprises revealed by modern science, one of the most remarkable is the discovery that the gut and the brain are in constant conversation. At first, this idea seems improbable. The brain, enclosed within the skull, is the seat of thought, memory, and perception. The gut, winding through the abdomen, is the organ of digestion and absorption. Yet, between these two distant regions runs a complex web of communication, often called the gut-brain axis. And at the center of this dialogue lies the microbiome. The microbes of the gut are not silent passengers, but active participants in a conversation that touches mood, emotion, stress, and even cognition. To explore this connection is to enter a realm where the boundaries between body and mind grow porous, and where tiny organisms influence the very experience of being human. The gut and the brain are linked physically through the vagus nerve, one of the longest nerves in the body. This nerve runs from the brainstem down into the chest and abdomen, carrying signals in both directions. Through the vagus, the gut can send messages to the brain, and the brain can, in turn, influence gut activity. For many years, scientists assumed this pathway was used only for basic functions like regulating digestion or controlling hunger. Now it is clear that the vagus nerve also carries signals produced by microbes, chemical messages that travel from the depths of the gut to the command center of the brain. In this way, microbes gain a voice in the inner conversation of the body, whispering through nerve fibers that reach directly into consciousness. Chemical signaling extends beyond the vagus nerve. The microbiome produces neurotransmitters, the very chemicals used by the brain to communicate. Certain gut bacteria generate serotonin, often called the happiness molecule, which influences mood and sleep. Others produce dopamine, linked with motivation and reward, or gamma-aminobutyric acid, known as GABA, which calms neural activity. Though most of these neurotransmitters act locally in the gut, they also send signals that influence the brain indirectly, either by traveling through the bloodstream or by activating neural pathways. The result is that microbial activity in the colon can, over time, affect how a person feels, how they respond to stress, and how they experience the world. This connection helps explain why emotions are often felt in the stomach. Anxiety may cause a tightening of the gut, while joy can create a sensation of lightness. These experiences are not merely poetic metaphors, but reflections of real communication. The gut sends signals to the brain about its state, and the brain responds by shaping perception. The microbiome, in turn, influences both sides of this conversation. By modulating gut activity and producing signaling molecules, microbes shape the messages that reach the brain. In this way, the gut becomes not only a digestive organ but an emotional one, a second brain influenced by microbial life. Stress provides a striking example of this relationship. When the brain perceives threat, it signals the gut through hormones and nerves, altering motility and secretion. These changes disrupt the microbial balance, sometimes reducing diversity or favoring stress-tolerant species. In return, the altered microbiome sends signals that amplify stress responses, creating a feedback loop. This cycle can influence mood, resilience, and mental health. Conversely, when the microbiome is balanced and diverse, it can dampen stress responses, releasing molecules that calm inflammation and restore equilibrium. The gut microbiome thus acts as both mirror and moderator of stress, reflecting the brain's state while also shaping it. Sleep is another domain influenced by this hidden dialogue. Microbes produce metabolites that affect circadian rhythms, the internal clock that governs sleep and wake cycles. Disruptions in the microbiome, such as those caused by irregular eating patterns or antibiotics, can alter these rhythms, leading to difficulties with rest. Conversely, when microbial communities are stable, they support the production of compounds that encourage restorative sleep. This subtle influence reveals how the microbiome extends beyond digestion into the regulation of fundamental aspects of life, such as rest and recovery. The gut-brain conversation is not limited to emotions and sleep, but extends to cognition and behavior. Animal studies have shown that changes in the microbiome can alter learning, memory, and social interaction. Germ-free mice, raised without any microbes, exhibit heightened anxiety and unusual behaviors, which are reversed when they are colonized with a healthy microbiome. While human studies are more complex, emerging evidence suggests that microbial composition can influence mental clarity, focus, and even risk for neurological conditions. These findings hint at a deep connection between microbial diversity and the brain's ability to function at its best. One way to picture this connection is as a vast telephone network, with countless microbial voices sending signals along different lines to the brain. Some speak through the vagus nerve, others through neurotransmitters, others still through immune pathways and hormones. The brain listens to these voices and incorporates them into its decisions, often without conscious awareness. The mood one feels upon waking, the appetite one experiences after a meal, the calm or agitation that arises in response to daily events, all are influenced, in part, by this microbial chorus. It is a dialogue so constant and subtle that we rarely notice it, yet it shapes the texture of everyday life. The microbiome also plays a role in shaping development of the nervous system itself. During infancy, when the brain is rapidly growing, Microbial signals influence how neural circuits form and mature. The absence of these signals can lead to altered brain development, reinforcing the idea that microbes are not peripheral to human life, but integral to its very foundation. Just as they train the immune system, they help sculpt the nervous system, guiding its early growth in ways that last a lifetime. There is humility in recognizing that human thoughts and emotions are not solely the products of the brain in isolation. They are influenced by organisms so small they can barely be seen. To realize this is to see that the mind is not separate from the body, nor the body separate from the environment it inhabits. The microbiome serves as a bridge, linking digestion, immunity, and the nervous system into a single web. Within this web, the brain does not stand alone at the top, but participates in an ongoing conversation, constantly listening and responding to the whispers of microbial life. This perspective does not diminish the uniqueness of human consciousness, but enriches it. To know that feelings of calm or unrest, clarity or confusion may be shaped by the microbiome is to appreciate the depth of interconnectedness that defines life. It is a reminder that health is not simply the absence of disease, but the presence of balance, a harmony between human cells and microbial companions. The microbiome speaks to the brain not in loud commands, but in gentle nudges, guiding emotions and thoughts in ways that often pass unnoticed yet are always present. In the end, communication with the brain is one of the most extraordinary functions of the microbiome, transforming our understanding of both digestion and emotion. It shows that the hidden organ is not confined to the gut but reaches into the realm of feeling and thought. To reflect on this connection is to realize that within us lies a partnership more intimate than we could have imagined, one in which microbes help shape the mind itself. They are not merely residents of the gut but participants in the human story, influencing the way we see, feel, and respond to the world. And so, as the hidden organ carries out its quiet work, it also weaves itself into the fabric of consciousness, reminding us that even the smallest forms of life can touch the largest mysteries of the mind. 9. The microbiome in early life. The story of the microbiome is inseparable from the story of human life itself, for its formation begins not long after birth and shapes health in ways that last across decades. To look at the earliest moments of life is to see how the hidden organ takes root, seeded by the environment, nourished by parents, and sculpted by circumstances that may seem ordinary, yet leave lasting imprints. The microbiome in early life is a foundation, a delicate architecture that influences immunity, metabolism, and even emotional development. Its establishment is as important as the growth of bones or the development of the nervous system, though it unfolds invisibly, carried by microbes so small they cannot be seen without magnification. Before birth, the infant's digestive tract is nearly free of microbes, sheltered within the protective environment of the womb. For many years, it was assumed that the womb was entirely sterile, though more recent studies suggest that tiny traces of microbes may already be present, passed from mother to child during pregnancy. Whether or not these early traces are significant, the true colonization begins at the moment of birth. As the infant passes through the birth canal, they are coated in maternal microbes from the skin and mucosal surfaces. These microbes form the first wave of settlers in the infant gut, beginning the process of building a community. Infants delivered by cesarean section, by contrast, encounter a different set of microbes, often those from the skin and hospital environment rather than the mother's birth canal. These early differences create variations in the microbiome that can persist for months or even years, demonstrating how the very process of birth leaves a microbial signature that influences development. Breastfeeding plays a central role in shaping the infant microbiome. Human milk is not only a source of nutrients, but also a complex medium designed to support microbial life. It contains beneficial bacteria directly transferred from mother to child, as well as special sugars known as human milk oligosaccharides. These sugars are indigestible by the infant but serve as nourishment for specific bacteria, particularly those in the genus Bifidobacterium. In this way, the mother's milk feeds not only the infant but also the microbes that will protect the infant's gut and train its immune system. Formula feeding, while providing essential nutrition, does not replicate this exact composition, and thus the microbial communities that develop may differ. The choice between breastfeeding and formula is shaped by many factors, but from the perspective of the microbiome, it is one of the first great influences on microbial development. As infants grow and begin to explore the world, their microbiomes diversify. Contact with parents, siblings, pets, and the broader environment introduces new microbes, each adding to the richness of the hidden organ. When solid foods are introduced, usually around 6 months of age, the gut microbiome undergoes a major transition. Species capable of breaking down complex plant fibers begin to thrive, and the ecosystem becomes more similar to that of an adult. These shifts are not random but structured, following predictable stages that correspond to diet and environment. By the age of 3, the microbiome is largely established, though it will continue to adapt throughout life. This early period is sometimes described as a window of opportunity when the foundation of microbial health is laid. The importance of this foundation becomes clear when one considers its impact on the immune system. Infants are born with immune systems that are functional but immature. Without guidance, these systems may overreact or fail to respond appropriately. The microbiome provides the lessons that guide immune maturation. It introduces antigens in controlled ways, teaching immune cells tolerance and balance. Infants who lack diverse microbial exposure may be more prone to allergies, asthma, or autoimmune conditions later in life. This observation has given rise to the hygiene hypothesis, which suggests that overly sterile environments in early life may hinder immune development. While cleanliness and medical care are essential, they must be balanced with exposure to the microbial world that trains immunity. The microbiome also influences metabolism from an early age. The way infants process nutrients, store energy, and regulate growth is affected by microbial composition. Studies have shown that differences in early microbiomes can influence the risk of obesity, diabetes, and other metabolic conditions in later life. These effects are not deterministic but probabilistic. The microbiome nudges development in certain directions, interacting with diet, genetics, and lifestyle. It is as though the microbiome writes the opening chapters of a book, setting the tone for the story that will follow, while later choices and environments write the rest. Birth circumstances and environmental exposures are not the only influences on the early microbiome. Antibiotic use is another powerful factor. While antibiotics are often necessary to combat infections, they also disrupt microbial communities, reducing diversity and sometimes allowing harmful species to flourish. Infants exposed to antibiotics may experience lasting changes in their microbiomes, though these can sometimes be mitigated by time, diet, and microbial resilience. This sensitivity highlights the fragility of the early microbiome, a system still establishing itself and easily shifted by external forces. The development of the microbiome in early life is not only biological but also ecological. It resembles the succession of a forest, beginning with pioneer species that prepare the soil, followed by waves of new arrivals that create balance and complexity. In the gut, early colonizers create conditions that allow later species to thrive, just as certain plants in a forest prepare the way for others. This ecological perspective helps explain why diversity and stability are key to a healthy microbiome. A forest with only one kind of tree is vulnerable to disease, while a forest with many species is resilient. Similarly, a microbiome with many types of microbes is more likely to withstand disturbance and support health. What makes the microbiome in early life especially profound is its quiet influence on the whole trajectory of human existence. These first microbial encounters shape not only digestion and immunity, but also emotional development. The gut-brain axis, already active in infancy, is influenced by microbial metabolites that affect mood and stress responses. Emerging research suggests that differences in early microbiomes may even shape patterns of behavior and resilience. Though these findings are still unfolding, they add to the growing recognition that the microbiome is not a passive companion but an active guide, shaping human development from the very beginning. In reflecting on this process, one cannot help but feel a sense of wonder. At birth, a new life enters the world, and with it, a new microbial community is born. This hidden organ grows alongside the infant, adapting, teaching, and supporting, often without recognition. It is a partnership that begins before words are spoken, before memories are formed, before consciousness itself emerges. To see the microbiome in early life is to see the roots of human health, planted in the soil of microbial diversity and nourished by the simplest acts of feeding, touching, and living. Thus, the microbiome in early life is both fragile and powerful, vulnerable to disturbance, yet essential for resilience. It is the unseen foundation upon which immunity, metabolism, and emotional well-being are built. To nurture it is to nurture the infant, and to recognize it is to understand that no human being comes into the world alone. Each arrives accompanied by a multitude of microbial companions, ready to begin the lifelong work of partnership. In this hidden beginning lies the quiet truth that health is not only inherited from parents, but also co-created with the invisible world of microbes that enter with the first breath and remain until the last. 10. The microbiome across cultures and environments. The microbiome, though hidden from sight, carries the imprint of culture, geography, and daily living. It is shaped not only by biology, but also by the rhythms of human society, reflecting the foods we eat, the places we inhabit, and the traditions we pass down through generations. To look at the microbiome across cultures and environments is to see how deeply it is tied to lifestyle, and how the diversity of human practices echoes as diversity within microbial communities. This hidden organ is not uniform across all people. It is as varied as language, music, or diet, forming patterns that tell the story of how people live and where they belong. One of the clearest examples of cultural influence on the microbiome lies in diet. Traditional diets rich in whole plants, roots, grains, and fibers nurture microbial communities that specialize in breaking down complex carbohydrates. These communities thrive in abundance, producing short-chain fatty acids that support gut health, immunity, and metabolism. By contrast, modern diets, often dominated by refined sugars, processed foods, and lower fiber intake, support a different microbial profile, sometimes less diverse and less resilient. Populations living in rural regions with traditional diets often show microbiomes that are richer in species than those of urban populations, reflecting the broader variety of plant matter and natural exposures present in their food. This difference is not small. It speaks to a divergence in microbial ecosystems that mirrors the divergence in cultural practices. Geography also leaves its mark. People living close to forests, farms, and natural environments are exposed to a broader range of environmental microbes, many of which become part of their microbial communities. Those in urban environments, surrounded by concrete, filtered air, and processed foods, encounter fewer sources of microbial diversity. Studies comparing populations in industrialized nations with those in traditional societies often reveal striking contrasts. For example, hunter-gatherer groups and rural farming communities tend to have gut microbiomes that contain species rarely found in urban populations, species that may play roles in digesting fibrous plants or protecting against inflammation. These differences highlight how the environment, both natural and built, shapes the inner landscapes of the microbiome. Cultural practices beyond diet also influence microbial composition. Methods of food preparation, fermenting, drying, cooking, or eating raw, introduce different microbial species into the gut. Fermented foods such as kimchi, sauerkraut, yogurt, and kefir carry live bacteria that can temporarily enrich the microbiome. In some cultures, fermented foods are daily staples, and their microbes may become recurring visitors within the digestive tract. In others, fermentation is rare, and the microbiome reflects this absence. Similarly, customs of physical contact, hygiene, and childcare affect microbial exposure. Infants carried close to their mothers, kissed and touched frequently, acquire different microbial communities than those raised in more sterile environments. Even the presence of pets alters the household microbiome, adding species from fur, soil, and outdoor spaces into the microbial pool of the family. Antibiotic use provides another cultural lens. In some regions, antibiotics are prescribed and used frequently, sometimes even without medical necessity. In others, their use is more restricted. The microbiome reflects these patterns, with populations exposed to frequent antibiotics, often showing reduced diversity and slower recovery after disruption. This cultural difference underscores how medical practices, while life-saving, carry long-term consequences for microbial communities. Similarly, sanitation practices differ across societies. Access to clean water and modern plumbing reduces exposure to harmful pathogens, but it also reduces contact with the broader microbial world. The balance between protection from disease and exposure to beneficial microbes is delicate, and different cultures navigate it in different ways. Migration provides a unique perspective on the microbiome's adaptability. When people move from one environment to another, their microbiomes shift to reflect the new culture and diet. Within a generation, the microbial profile of a family may resemble that of the host country more than that of the homeland. This rapid change shows how flexible the microbiome can be, but it also raises questions about what may be lost in the process. Certain species associated with traditional diets may disappear when families adopt new lifestyles, potentially altering health outcomes. These changes mirror the broader transitions of culture itself, where traditions adapt, evolve, and sometimes fade. The microbiome carries this cultural narrative at the microscopic level, recording it in shifts of species and genes. The comparison of microbiomes across cultures has also revealed the existence of a core microbiome groups of microbial species found in nearly all humans, regardless of location. These core species provide essential functions such as fermenting fibers, synthesizing vitamins, and regulating immunity. Yet beyond this shared foundation lies a vast realm of variability, unique to each culture and environment. The core ensures basic survival, but the surrounding diversity may determine resilience, adaptability, and the subtler aspects of health. This dual structure, shared foundation, and cultural variation parallels human life itself, where universal biological needs are met within the unique context of cultural expression. The microbiome also reflects the impact of modernization and industrialization. Traditional societies often maintain high microbial diversity, while industrialized societies show reduced variety. This decline has been linked to increases in certain chronic conditions, such as allergies, asthma, and autoimmune diseases, suggesting that the loss of microbial diversity may carry hidden costs. Some researchers have described this as the disappearing microbiome phenomenon, a quiet erosion of diversity as lifestyles move further from traditional patterns. Yet even within modern societies, subcultures that maintain diverse diets, regular contact with nature, and traditions of fermentation show microbiomes that retain greater richness. This suggests that cultural choices can, to some extent, preserve microbial diversity even in industrialized settings. Looking at the microbiome across cultures also highlights the deep interconnectedness between humans and the natural world. In traditional farming societies, contact with soil, animals, and plants introduces microbes that enrich the gut. In industrialized cities, where such contact is minimized, this link weakens. The microbiome thus mirrors humanity's relationship with the environment, recording how much or how little we remain tied to the earth. It is as though the hidden organ is a living archive, preserving the traces of cultural practices, environmental exposures, and ecological connections. This perspective also invites humility. There is no single ideal microbiome, no one-size-fits-all composition that defines health. Instead, there are many paths to microbial balance, each shaped by culture and environment. A rural farmer in one part of the world may host species adapted to digesting wild plants, while an urban resident may host species adapted to processed foods. Both microbiomes perform essential functions, though they do so in different ways. What matters is not conformity but resilience, the ability of the microbiome to adapt, recover, and maintain balance. This resilience is supported by diversity, and diversity, in turn, is supported by cultural and environmental richness. In reflecting on the microbiome across cultures, one sees how human life is mirrored at the microbial level. The foods we grow, the ways we cook, the environments we inhabit, and the traditions we keep are all written into the hidden organ within us. Each culture carries not only stories, songs, and languages, but also microbial signatures, inherited from ancestors and passed on to children. These signatures remind us that health is not only a matter of biology, but also of culture, that the hidden organ reflects not just the body, but the life lived within it. To honor the microbiome is, in part, to honor cultural diversity itself, for both are expressions of the same truth, that life thrives in many forms, each adapted to its environment, each carrying its own kind of wisdom. 11. The microbiome and disease. The microbiome, though often a source of resilience and balance, is not immune to disruption. Like a forest that can shift from harmony to imbalance when disturbed, the microbial communities within the body can also falter, leading to states that are less stable, less diverse, and less supportive of health. This condition, often referred to as dysbiosis, does not always manifest immediately as illness, but it creates an environment in which disease is more likely to arise. To understand the microbiome and disease is to recognize how tightly health is bound to microbial balance, and how easily this balance can be unsettled by diet, stress, medication, or environmental change. The microbiome is both guardian and vulnerability. When it is strong, it protects. When it is disturbed, it may open the door to illness. One of the clearest examples of this connection lies in the digestive system itself. Conditions such as irritable bowel syndrome, inflammatory bowel disease, and chronic diarrhea have been linked to shifts in the gut microbiome. In these cases, the normal balance of microbial species is altered, with beneficial bacteria declining and potentially harmful ones rising in abundance. This imbalance can lead to inflammation of the gut lining, increased permeability of the intestinal barrier, and altered immune responses. The gut, once a place of cooperation between host and microbes, becomes a site of tension and disruption. Patients with these conditions often show reduced microbial diversity, a sign that the forest within has lost some of its stabilizing species, leaving it vulnerable to swings and overgrowths that disturb its equilibrium. Obesity and metabolic disorders provide another striking illustration. Research has shown that individuals with obesity often host microbiomes that differ in composition from those of lean individuals. Certain microbial profiles are more efficient at extracting energy from food, which may contribute to weight gain, while diet and lifestyle remain central drivers of obesity. The microbiome plays a role in modulating how calories are processed and stored. Moreover, microbial products can influence hormones that regulate appetite and metabolism, shaping the body's energy balance in subtle ways. This connection suggests that the microbiome is not merely a passenger in metabolic health, but an active participant in the regulation of weight, insulin sensitivity, and fat storage. Dysbiosis in this context can tip the scales toward disease. Allergies and autoimmune conditions also reflect the role of the microbiome in disease. The immune system depends on microbial training during early life to distinguish between harmful and harmless stimuli. When this training is incomplete or disrupted, the immune system may overreact to benign substances such as pollen, dust, or food proteins, leading to allergies. In autoimmune diseases, the immune system may even turn against the body's own tissues. Dysbiosis has been implicated in conditions such as type 1 diabetes, multiple sclerosis, and rheumatoid arthritis, where alterations in microbial communities appear to influence immune regulation. While microbes do not cause these diseases directly, their absence or imbalance may remove the signals that would otherwise keep immune activity balanced and measured. The skin, too, shows how the microbiome can shift toward disease. Eczema, acne, and other inflammatory skin conditions have been linked to changes in microbial populations on the skin's surface. Healthy skin hosts a diverse community of microbes that defend against pathogens and maintain balance. When this community is disrupted, harmful bacteria may proliferate, or the immune system may react in ways that produce inflammation. Here again, diversity and balance are protective, while imbalance leaves room for disease. The influence of the microbiome extends even to the lungs and respiratory system. Asthma and chronic obstructive pulmonary disease have been associated with changes in the airway microbiome. Children who grow up in environments with reduced microbial exposure, such as overly sanitized homes or urban settings with limited contact with nature, appear to have higher rates of asthma. This pattern suggests that microbial diversity in the airways plays a role in training the immune system to respond calmly to harmless particles. Without this microbial guidance, the immune system may interpret ordinary exposures as threats, triggering chronic inflammation of the airways. Mental health is another frontier where the microbiome and disease intersect. The gut-brain axis shows how microbial signals influence mood, stress, and cognition. Dysbiosis has been associated with depression, anxiety, and other psychiatric conditions, though the exact mechanisms remain under study. It is believed that altered microbial communities may disrupt the production of neurotransmitters and inflammatory signals, influencing brain function. While mental health cannot be reduced to the microbiome alone, the recognition that microbial imbalance may contribute adds a new dimension to understanding these conditions. It also reinforces the idea that health is not confined to separate compartments of the body, but is shaped by interconnected systems in which the microbiome plays a pivotal role. Antibiotic use illustrates vividly how the microbiome can be disrupted and how disease may follow. Antibiotics are powerful tools that save lives by killing harmful bacteria, but they do not discriminate perfectly. Alongside pathogens, they also eliminate beneficial species, sometimes leaving behind empty niches that harmful bacteria can exploit. This disruption can lead to conditions such as Clostridioides difficile infection, a disease marked by severe diarrhea and inflammation that often arises after antibiotic treatment. Here, the absence of protective microbes allows a dangerous species to flourish unchecked. Recovery often depends on restoring microbial diversity, whether through natural regrowth, diet, or medical interventions designed to repopulate the gut. Cancer research has also begun to reveal links between the microbiome and disease. Certain microbial profiles are associated with increased risk of colorectal cancer, possibly through the production of compounds that damage DNA or promote inflammation. Conversely, Some microbes appear to protect against cancer, producing compounds that inhibit tumor growth or enhance immune surveillance. Moreover, the microbiome can influence how individuals respond to cancer therapies, including immunotherapy. These findings highlight the dual nature of the microbiome in disease. It can both contribute to risk and provide pathways to resilience, depending on its composition and balance. Despite these associations, it is important to recognize that the microbiome does not act in isolation. Disease emerges from a complex interplay of genetics, environment, lifestyle, and microbial communities. The microbiome is one piece of this puzzle, albeit a powerful one. To view it as a hidden organ is to acknowledge its central role, but also to see that its influence depends on context. A diverse and balanced microbiome supports health. A disrupted one can tilt the balance toward disease. The line between protection and vulnerability is delicate, shaped by choices, exposures, and chance events that ripple through the microbial ecosystem. In reflecting on the microbiome and disease, one is struck by both fragility and resilience. The microbial forest within can be disturbed by antibiotics, poor diet, or stress, yet it can also recover, regrowing its diversity and restoring balance. Disease arises not from microbes alone, but from the collapse of harmony, the tipping of scales that leaves the body vulnerable. To care for the microbiome is not simply to avoid disease, but to nurture resilience, ensuring that the hidden organ remains strong enough to support health across the unpredictable turns of life. Thus, the microbiome and disease are inseparable aspects of the same story. The hidden organ sustains health through balance, but when balance is lost, it can also become a site of vulnerability. It is both shield and weakness, both protector and potential accomplice in illness. To understand disease through the lens of the microbiome is to see that health is not static but dynamic, maintained through constant dialogue between host and microbes. It is a reminder that within the body, as in the natural world, balance is fragile, harmony precious, and diversity the key to resilience. 12. The skin microbiome. The skin, spread across the body as its largest organ, is both shield and frontier. It stands at the border between the internal world of the body and the external world of air, water, soil, and countless living forms. Though often imagined as a simple protective covering, the skin is alive with its own ecosystems, populated by microbial residents that form what is known as the skin microbiome. To look closely at the skin is to see not emptiness, but a living landscape of microbes, working quietly to maintain balance, defend against threats, and cooperate with the body in subtle ways. The skin microbiome is less dense than that of the gut, but no less significant, for it helps shape the delicate harmony of health at the body's surface. The skin is not uniform, and this diversity of terrain creates multiple habitats for microbial life. Dry regions, such as the forearms and legs, host species adapted to scarcity, microbes that can endure limited moisture and nutrients. Moist regions, such as beneath the arms or between toes, harbor dense populations of bacteria and fungi that thrive in warmth and humidity. Oily regions, like the forehead and upper back, provide lipids that nourish microbes capable of breaking down sebum, the oily secretion of glands in the skin. Each of these niches supports a distinct community, and together they form a mosaic of microbial life spread across the body. This patchwork is not random but patterned, shaped by physiology, environment, and daily habits. The skin microbiome plays an essential role in defense. The skin is constantly exposed to potential invaders, from environmental microbes to pathogens carried by touch. Beneficial microbes help guard the surface by occupying space and consuming available nutrients, leaving little room for harmful species to take hold. Many produce antimicrobial compounds that directly inhibit the growth of rivals, acting as natural protectors of their chosen niche. Some even communicate with immune cells beneath the skin, reinforcing defenses and modulating responses. In this way, the skin microbiome becomes part of the body's protective barrier, not only physical but biological. The shield of the skin is thus more than layers of keratin. It is also a living layer of microbial guardianship. Balance is key to this protection. When microbial diversity is stable, the skin remains resilient. But when balance is disrupted by antibiotics, harsh cleansers, or underlying health conditions, the result may be vulnerability. In such cases, harmful microbes can flourish, or the immune system may overreact, producing inflammation. Conditions such as eczema, acne, and psoriasis often reflect disturbances in the skin microbiome. These are not caused by single pathogens, but by shifts in community composition, where protective species decline and opportunistic ones expand. Just as a forest becomes fragile when its diversity is reduced, the skin loses resilience when its microbial community is narrowed or unbalanced. The relationship between the skin microbiome and the immune system is especially deep. Immune cells in the skin are constantly sampling signals from microbes, learning to distinguish harmless residents from dangerous intruders. Beneficial microbes reinforce tolerance, preventing unnecessary inflammation, while also training immune cells to remain ready for genuine threats. Without these microbial signals, the immune system may become hypersensitive, reacting to harmless stimuli such as pollen, soaps, or minor abrasions. This partnership ensures that the immune system is neither too passive nor too aggressive, a balance critical to the skin's function as both barrier and interface with the outside world. Daily life exerts a constant influence on the skin microbiome. Washing, clothing, cosmetics, climate, and even the touch of others all leave traces. Soap and water remove surface microbes, though many quickly return as they regrow from deeper layers or from surrounding environments. Clothing creates microclimates of warmth and moisture that shape microbial populations. Cosmetics and lotions alter the chemical composition of the skin surface, favoring some species over others. Climate, too, plays a role. Humid environments nurture different microbial communities than arid ones. Even geography leaves a mark, as people living in different regions host skin microbiomes, subtly tuned to local conditions. This responsiveness reflects the adaptability of microbial life, its capacity to shift with the rhythms of human activity. The skin microbiome also carries echoes of human individuality. Just as fingerprints are unique, so too are microbial patterns on the skin. No two people share exactly the same microbial composition, though families and close companions often show similarities, shaped by shared environments and contact. This individuality has even been studied in forensic science, where skin microbes left behind on objects can sometimes be traced back to their owner. While these applications are technical, they highlight the intimate connection between skin microbes and personal identity, a connection that forms silently each day as we move through the world. Interestingly, the skin microbiome is not static but evolves across the lifespan. Infants are born with skin that is quickly colonized by microbes from the mother and environment. These early communities are simple but expand as the child grows, adapting to new exposures. Adolescence, with its hormonal changes and increased production of oils, reshapes the skin microbiome dramatically, often contributing to conditions like acne. Adulthood brings stability, though daily habits continue to influence composition. In old age, changes in skin structure. The skin microbiome, like the skin itself, reflects the stages of life, carrying the imprint of growth, maturity, and aging. The skin microbiome also demonstrates the profound interconnectedness of the body. While it resides on the surface, it communicates with deeper systems, influencing immunity, inflammation, and even responses to environmental stress. It connects the body with the external world, serving as a mediator between human physiology and the ecosystems that surround us. Every touch, every breath of air, every surface we encounter introduces microbes that interact with those already present. The skin microbiome is thus not isolated but part of a constant exchange, a reminder that the boundary between self and environment is porous and alive. To imagine the skin microbiome is to picture a living tapestry spread across the body, each region woven with its own colors and patterns, each thread representing species that have learned to coexist. It is a tapestry that shifts subtly each day, responding to climate, touch, and care, yet retaining its fundamental integrity. Like a shield woven from living fibers, it protects, balances, and communicates, sustaining the quiet harmony of the body. Though invisible, it is as real and as vital as the skin we can see, a hidden organ layered upon the visible one. In the end, the skin microbiome teaches a lesson of coexistence. The skin is not a sterile barrier but a community, where countless species live in balance with human cells. Health depends not on eliminating these microbes, but on sustaining their diversity and harmony. They are companions at the surface of our being, silent allies who guard, guide. To reflect on the skin microbiome is to realize that even the most visible part of the body holds hidden depths, that protection comes not from exclusion but from partnership, and that within the touch of skin lies the quiet presence of life, too small to see, yet too important to forget. 13. Other microbial niches. While the gut and the skin hold the largest and most studied microbial communities, the hidden organ of the microbiome extends far beyond these familiar territories. The human body is a vast landscape, and microbes have found niches in places that may seem unlikely. Yet each of these sites hosts its own delicate ecosystem. The mouth, the lungs, the reproductive tract, the eyes, and the ears all contain microbial life, each community adapted to its unique environment. To explore these smaller but vital niches is to see how thoroughly the microbiome permeates the human body, filling spaces with life too small to see but essential for balance, defense, and function. The mouth is among the most densely populated microbial habitats outside the gut. Constantly exposed to food, drink, and breath, it provides moisture, warmth, and abundant surfaces for microbes to colonize. The tongue, with its rough texture, offers ridges where bacteria can anchor, forming communities that persist despite the constant flow of saliva. The teeth provide hard surfaces where biofilms, known as dental plaque, develop. Within these biofilms, bacteria form layered communities protected by a matrix they secrete, allowing them to resist removal and interact closely. Some species aid in breaking down food, while others maintain balance by competing with potential pathogens. Yet when balance is disrupted by poor oral hygiene, sugary diets, or illness, the oral microbiome can shift toward harmful forms, contributing to cavities, gum disease, and systemic conditions linked to oral health. Thus, the mouth is not only the gateway to digestion, but also a vital microbial niche whose stability influences the entire body. The lungs, once thought to be sterile, are now recognized as another site of microbial life. The microbial populations here are sparse compared to those in the gut or mouth, yet they play important roles in shaping immunity and maintaining balance in the respiratory tract. The lungs are constantly exposed to the outside world through inhalation, bringing in particles, pollutants, and microbes with each breath. Beneficial residents help train the immune system to distinguish between harmless particles and dangerous invaders, reducing the likelihood of overreactions that lead to chronic inflammation. When the lung microbiome is disturbed, however, conditions such as asthma, chronic obstructive pulmonary disease, and infections become more likely. Though smaller in scale, the lung microbiome is a reminder that even the most delicate and vital organs rely on microbial balance for health. The reproductive tract presents yet another specialized microbial ecosystem. In women, The vaginal microbiome is dominated by lactobacillus species, which produce lactic acid and maintain a slightly acidic environment. This acidity acts as a natural defense, discouraging harmful bacteria and fungi from thriving. The balance of this microbiome is crucial, for disturbances can lead to infections, discomfort, or increased vulnerability to disease. In men, the reproductive tract hosts its own communities, less studied but equally important, influencing reproductive health and potentially interacting with partners' microbiomes. These intimate microbial ecosystems are deeply tied to reproduction and sexual health, reflecting how the microbiome extends into the most personal aspects of human life. The eyes, often imagined as too delicate for microbial presence, also host their own communities. Though sparse compared to the skin or gut, the microbes of the eye live on the conjunctiva and within the tear film, constantly bathed in protective fluids. These microbes help guard against infection, maintaining balance in a region that is exposed to air, light, and environmental particles. Disruption of the ocular microbiome has been linked to conditions such as conjunctivitis or dry eye disease, suggesting that even here, microbial balance is necessary for comfort and health. The presence of life in such a sensitive organ is a reminder that the microbiome spares no surface, filling even the quiet landscape of the eye with unseen guardians. The ears, too, contain their own microbial populations. Within the ear canal, microbes adapt to a dark, slightly waxy environment. Earwax itself, or cerumen, provides nutrients while also protecting against pathogens. The microbes of the ear help maintain this balance, contributing to the ear's natural defenses. As with other niches, imbalance can lead to infections or discomfort, but when in harmony, these microbes remain unnoticed, performing their roles in silence. Together, these niches form a vast and interconnected web. Though the microbes of the mouth, lungs, reproductive tract, eyes, and ears differ in species and function, they all share a common theme. While imbalance opens the door to disease, each niche interacts not only with its immediate environment but also with the body as a whole. Oral health influences cardiovascular health, lung microbes influence immune regulation, reproductive tract microbes influence fertility and susceptibility to infection. These interactions reveal that no microbial community is isolated. Instead, they form a network of ecosystems, linked by the circulation of blood, the signaling of hormones, and the communication of the immune system. It is striking to realize how adaptable microbes are to such varied conditions. Some thrive in the acidic environment of the vagina, others in the waxy folds of the ear, others still in the moist and shifting terrain of the mouth. Each has found a way to coexist with human tissues, not as invaders but as residents. Their presence is a testament to the resilience and versatility of life, able to flourish in every corner where nutrients and shelter are available. Just as life on Earth has filled oceans, deserts, and mountain tops, microbial life has filled every corner of the human body, forming a parallel ecology within us. These niches also reflect individuality. Just as no two fingerprints are the same, no two people share identical microbial communities in these smaller habitats. The microbes of the mouth, lungs, or reproductive tract are shaped by diet, lifestyle, environment, and personal history. Families and close companions may share similarities, yet each microbiome remains unique, a personal signature written in invisible life. This individuality may help explain why people differ in susceptibility to certain conditions or in their responses to treatments. The microbiome, in all its niches, personalizes health in ways still being uncovered. In reflecting on these other microbial niches, one sees the breadth of the hidden organ. It is not confined to the gut or the skin, but spread across the entire body, inhabiting places we rarely think of as microbial. It fills every surface exposed to the world, every fold where moisture and nutrients allow life to persist. These niches are small, yet their influence is profound, shaping health at the boundaries of the body. They remind us that the microbiome is not one organ but many interlinked communities, together forming a hidden system that sustains life. To imagine the microbiome as a whole is to picture a constellation of niches, each with its own stars, each contributing light to the larger sky. The mouth, the lungs, the reproductive tract, the eyes, the ears all are part of this constellation, linked together in patterns that reflect both biology and environment. Each niche is quiet, invisible, and easily overlooked, yet each plays a part in the harmony of health. The hidden organ is not only vast in its total population, but also intricate in its distribution, reaching into every corner of the body with unseen but essential life. 14. The microbiome and metabolism. The story of metabolism is often told as the story of human cells, transforming food into energy, converting sugars, fats, and proteins into the fuel that powers life. Yet behind this familiar narrative lies another, quieter one. The role of the microbiome in shaping how energy is extracted, stored, and used. The microbes within us are not passive observers of nutrition but active participants in metabolism, guiding the way the body responds to food and influencing processes that extend far beyond digestion. To explore the microbiome and metabolism is to see that energy balance, weight regulation, and even the risk of chronic disease are deeply tied to the hidden organ that thrives in the gut and beyond. At the most basic level, microbes assist in breaking down substances that human enzymes cannot digest. Fibers, resistant starches, and other complex carbohydrates are fermented by gut bacteria into short-chain fatty acids such as butyrate, acetate, and propionate. These molecules are more than energy sources. They act as regulators, signaling to host cells how to use nutrients. Butyrate nourishes the cells lining the colon, helping maintain barrier integrity. Acetate and propionate circulate through the bloodstream, influencing the liver and muscles in their handling of glucose and fat. These subtle signals adjust metabolism in ways that ripple through the entire body, showing that microbial byproducts are not waste but messages, guiding how energy is managed. The microbiome also shapes how the body stores fat. Certain microbial profiles are more efficient at extracting calories from food, leading to greater energy harvest. Studies comparing lean and obese individuals have found differences in microbial composition, with some species associated with increased energy extraction and fat storage. In experiments with mice, transferring the microbiome of obese animals into lean ones caused the recipients to gain weight, even when their diets remained unchanged. These findings suggest that microbes can tilt the balance of metabolism, nudging the body toward energy storage or energy expenditure. While human metabolism is shaped by many factors, including genetics and lifestyle, the microbiome adds another layer of influence, one that may explain why people respond differently to the same foods. Hormones that regulate appetite and energy balance are also influenced by microbial activity. Short-chain fatty acids, produced by fermentation, stimulate the release of hormones such as peptide YY and glucagon-like peptide 1, which promote feelings of fullness and regulate insulin secretion. By modulating these hormones, microbes participate in the control of appetite and glucose metabolism. This partnership blurs the line between digestion and signaling, showing that the gut microbiome does not merely process food but also helps govern the brain's perception of hunger and satiety. Every meal becomes part of a dialogue between food, microbes, and the endocrine system, shaping energy intake in subtle but powerful ways. The microbiome's role in metabolism also extends to the processing of lipids and bile acids. The liver produces bile to emulsify fats, but once released into the intestine, bile acids are modified by microbes into secondary bile acids. These compounds influence fat absorption, cholesterol levels, and even the activity of receptors that regulate metabolism throughout the body. In this way, Microbes act as chemists, transforming bile into molecules that guide host physiology. Disturbances in this process may contribute to metabolic disorders, linking microbial balance to cholesterol, regulation, and cardiovascular health. Insulin sensitivity, a cornerstone of metabolic health, is similarly influenced by microbial activity. Some microbial products improve insulin signaling, making it easier for cells to take up glucose from the blood. Others promote inflammation, which can interfere with insulin action and contribute to conditions such as type 2 diabetes. The balance between these effects depends on the composition of the microbiome. A diverse and balanced microbial community tends to support insulin sensitivity, while dysbiosis may tilt metabolism toward resistance and dysfunction. This interplay highlights the microbiome as a key factor in the body's ability to manage blood sugar and maintain metabolic equilibrium. Beyond the gut, the microbiome influences metabolism indirectly through its interactions with the immune system. Low-grade inflammation, often linked with obesity and metabolic syndrome, can arise when microbial balance is disturbed. Certain bacterial components, if they cross a weakened intestinal barrier, may trigger immune responses that promote inflammation. This state of chronic subtle immune activation alters metabolism, affecting fat storage, insulin action, and energy use. In this way, the microbiome acts as both gatekeeper and regulator, determining whether the immune system supports or hinders metabolic health. The adaptability of the microbiome underscores its influence on metabolism. Changes in diet can reshape microbial communities within days, altering the production of metabolites that affect energy balance. A fiber-rich diet fosters microbes that generate beneficial short-chain fatty acids, supporting insulin sensitivity and satiety. A diet dominated by refined sugars and fats, by contrast, encourages microbes that produce compounds associated with inflammation and metabolic dysfunction. These shifts demonstrate how dietary choices act not only on human physiology, but also on the microbial organ that mediates metabolism. The foods one eats become, in effect, messages to the microbiome, which then interprets them and responds in ways that shape health. The relationship between microbiome and metabolism also sheds light on why weight management is so complex. Two people may follow the same diet and lifestyle yet experience different outcomes, in part because their microbiomes process food differently. One person's microbes may be adept at extracting every possible calorie, while another's may favor fermentation pathways that produce fewer usable calories. These individual differences remind us that metabolism is not solely dictated by willpower or environment, but also by the hidden ecology within. The microbiome personalizes metabolism, making each body's response to food unique. While much of this influence supports health, imbalance can contribute to disease. Dysbiosis, marked by reduced diversity or dominance of harmful species, has been linked to obesity, diabetes, fatty liver disease, and cardiovascular conditions. These associations are not absolute, but they highlight the microbiome as a central player in the network of factors that shape metabolic health. It is not merely a background contributor but an active organ, constantly adjusting signals that determine how energy is used and stored. The resilience of the microbiome, however, offers hope. With time and support, it can often recover from disruptions, restoring metabolic balance. Reflecting on the microbiome and metabolism reveals a picture of profound interdependence. The body provides food and shelter for microbes, and in return, microbes guide how that food becomes energy. They transform indigestible fibers into fuels, regulate hormones that control appetite, shape insulin sensitivity, and influence fat storage. They act as hidden regulators, constantly adjusting the metabolic system to maintain balance. This partnership is ancient, forged through millions of years of coevolution, and it continues, each day, in the quiet depths of the gut. To imagine the microbiome's role in metabolism is to envision a symphony of signals, each note representing a microbial metabolite or hormone, each harmony a balance of energy and storage. It is a symphony conducted not by human will alone, but by the cooperation of countless microbial voices. Health emerges when the symphony is balanced, when the notes blend smoothly into a whole. Disease arises when the harmony is disrupted, when certain voices grow too loud or others fall silent. In this metaphor lies the truth. Metabolism is not a solitary act of the body, but a shared performance, guided by the hidden organ that lives within. 15. Antibiotics and modern challenges. The discovery of antibiotics was one of the most transformative achievements in the history of medicine. For the first time, humanity had tools powerful enough to combat bacterial infections that had plagued civilizations for centuries. Illnesses once considered fatal became treatable, and entire populations were spared from epidemics that would have cut lives short. Yet alongside this triumph came unintended consequences. Antibiotics, though life-saving, are not selective instruments. They kill harmful bacteria, but also sweep away beneficial species, leaving the microbiome disrupted and vulnerable. In exploring antibiotics and modern challenges, one sees how this essential medical tool also reshapes the hidden organ, altering the delicate ecosystems that support health in ways both temporary and lasting. When antibiotics enter the body, they move swiftly through tissues, targeting bacterial cells. But these drugs cannot always distinguish between pathogens and commensals, between invaders and allies. The gut microbiome, with its immense diversity, is particularly affected. Within days of beginning an antibiotic course, microbial populations may decline sharply, with some species nearly eliminated. Diversity shrinks, and the balance of communities shifts. While the body often recovers after treatment ends, the regrowth may not restore the original state. Some species fail to return, leaving gaps in the ecosystem. These gaps can persist for months or years, subtly altering digestion, immunity, and metabolism. One of the most dramatic examples of antibiotic disruption is the infection caused by Clostridioides difficile, a bacterium that can overgrow when other gut microbes are diminished. Normally, the diverse microbiome keeps C. difficile in check, but after antibiotics clear away competitors, this species may flourish, releasing toxins that cause severe diarrhea and inflammation. Treatment often requires further antibiotics, creating a cycle of disruption and vulnerability. In recent years, approaches such as fecal microbiota, transplantation, restoring diversity by introducing healthy microbial communities, have shown promise in treating recurrent infections. This therapy highlights the recognition that balance, rather than eradication, is the key to resilience. Beyond acute infections, the widespread use of antibiotics raises broader concerns. Overprescription, incomplete courses, and use in agriculture have contributed to the rise of antibiotic resistance, where bacteria evolve defenses against the very drugs designed to destroy them. This challenge threatens not only the treatment of infections but also the microbiome itself, as resistant strains may persist and alter microbial communities. The hidden organ becomes a reservoir where resistance genes circulate, spreading between species and across populations. This reality underscores the interconnectedness of medical practices, microbial ecology, and global health. Antibiotics also leave subtler imprints on the microbiome, especially when exposure occurs early in life. Infants given antibiotics in their first years may experience shifts in microbial composition that influence immunity and metabolism for years to come. Some studies suggest links between early antibiotic use and higher risks of allergies, asthma, and obesity, though the connections are complex and influenced by many factors. These associations reflect how fragile the developing microbiome is, how its balance can be tipped during its formative stages, and how those shifts echo across a lifetime. The promise of antibiotics is immense, but so too is the responsibility to use them wisely, particularly in the earliest years of life. Modern challenges extend beyond antibiotics themselves to the environments created by modern living. Urbanization, processed foods, sanitized surroundings, and reduced contact with soil and animals all contribute to a narrowing of microbial diversity. The microbiome thrives on exposure, on constant interactions with microbes from food, water, and the natural world. In modern environments, these exposures are fewer, and the hidden organ grows less diverse. Antibiotics amplify this narrowing, accelerating the decline of species that once flourished. What emerges is not a single catastrophic loss, but a gradual erosion of richness, a thinning of the microbial forest within. This erosion is linked to the rise of chronic conditions in industrialized societies. As diversity decreases, resilience weakens, making the microbiome more vulnerable to disturbances. Allergies, autoimmune conditions, and metabolic disorders appear more frequently, suggesting that the loss of microbial partners plays a role. While these conditions have many causes, the microbiome acts as an unseen mediator, translating lifestyle, diet, and medical practices into health outcomes. Antibiotics, when used frequently, may tip this balance further, compounding the effects of modern living. Yet the story is not one of despair, but of awareness. Understanding the impact of antibiotics on the microbiome has led to efforts to preserve microbial health. Physicians increasingly weigh the necessity of prescriptions, seeking to use antibiotics only when truly needed. Researchers explore ways to support recovery after treatment, whether through diet, probiotics, or microbial restoration therapies. Public health initiatives encourage stewardship, reminding us that antibiotics are precious resources whose use shapes not only individual outcomes, but the microbial landscapes of entire populations. The recognition of the microbiome as a hidden organ has reframed antibiotics not only as weapons against disease, but also as forces that reshape ecosystems within. Modern challenges also bring opportunities for innovation. Advances in sequencing technologies allow scientists to map microbial changes after antibiotics with remarkable precision, identifying which species are lost and how communities reorganize. These insights open the possibility of designing targeted therapies that spare beneficial microbes while attacking pathogens. New generations of antibiotics, probiotics, and microbiome-based treatments may one day restore balance more precisely, reducing the collateral damage of broad-spectrum drugs. In this vision, medicine evolves from blunt eradication to careful cultivation, treating infections while sustaining the ecosystems that support life. Reflecting on antibiotics and modern challenges is a reminder of the complexity of health. The human body is not a solitary organism, but a partnership with trillions of microbial companions. Antibiotics disrupt this partnership, sometimes necessarily, sometimes excessively. The challenge of modern medicine is to balance the undeniable benefits of these drugs with the need to protect the hidden organ. This balance requires not only scientific knowledge but also cultural awareness, shaping practices in homes, hospitals, and farms alike. It is a challenge that extends beyond individuals to communities and across generations. For the microbiome is both personal and collective. In the end, antibiotics reveal the dual nature of the microbiome, its strength and its fragility. The hidden organ can withstand disruption and regrow, but only to a point. Excessive use or repeated disturbance can erode its diversity, leaving it weakened. To value the microbiome is to treat antibiotics with respect, to see them not as casual remedies, but as powerful tools to be used with care. The modern challenge is not to abandon antibiotics, but to wield them wisely, recognizing that every dose reverberates through the microbial forest within. By preserving balance, we preserve not only the hidden organ but also the resilience of human health itself. 16. Restoring balance. When the microbiome is disrupted, The body often feels the consequences in subtle but persistent ways. Digestion may falter, immunity may become more reactive, and energy may fluctuate. This state of imbalance, known as dysbiosis, is not always permanent. Just as a forest can recover after fire or drought, the microbial communities of the body often possess resilience, capable of regrowth and renewal when conditions allow. Restoring balance is therefore less about drastic intervention and more about gentle cultivation, supporting the return of diversity, harmony, and cooperation among the microbes that form the hidden organ. It is a process shaped by diet, environment, and lifestyle, unfolding quietly within, often unnoticed, yet vital for long-term health. One of the most natural ways balance is restored is through diet. Microbes thrive on what the host consumes, and different foods nourish different communities. Diets rich in plant fibers provide the raw material for fermentation, supporting species that produce beneficial short-chain fatty acids. These molecules, in turn, strengthen the gut lining, reduce inflammation, and support immune regulation. The simple act of eating a variety of vegetables, fruits, legumes, and whole grains becomes a way of feeding not just oneself, but also the trillions of microbial companions within. Diversity in diet fosters diversity in microbes, creating resilience, much like a varied ecosystem resists collapse. By contrast, diets dominated by processed foods and refined sugars tend to narrow microbial communities, favoring species that thrive on simplicity but offer fewer benefits. Thus, diet acts as both gardener and architect of microbial balance, shaping the hidden organ with every meal. Fermented foods represent another gentle support for restoring balance. Across cultures, fermentation has long been used to preserve food and enhance flavor. Foods such as yogurt, kefir, sauerkraut, kimchi, and miso contain live microbes that, when consumed, pass through the digestive tract. Though many of these microbes do not permanently colonize the gut, their presence provides temporary enrichment, introducing new species and interacting with resident communities. They may also produce compounds that influence immunity and digestion while in transit. The tradition of fermentation, woven into so many cuisines, reflects an ancient partnership between humans and microbes, one that continues to offer benefits in the modern age. Prebiotics, substances that nourish beneficial microbes, also play a role in restoring balance. Unlike probiotics, which introduce live organisms, prebiotics provide the nutrients those organisms need to flourish. Many prebiotics are plant fibers that escape digestion in the small intestine and reach the colon intact, where microbes ferment them into useful metabolites. Garlic, onions, bananas, asparagus, and chicory are rich in such compounds. By including these foods in the diet, individuals can gently encourage beneficial microbial species to grow, strengthening the stability of the ecosystem within. This support reflects the principle that restoration is not only about adding new members, but also about nurturing those already present, allowing them to thrive. Beyond food, the environment itself contributes to microbial restoration. Contact with nature exposes the body to a broader range of microbes from soil, plants, and animals. Walking through forests, tending gardens, or simply spending time outdoors introduces organisms that may supplement or stimulate microbial diversity. In traditional societies, where daily life involves close contact with the natural world, microbiomes tend to be richer and more varied. In modern urban environments, this exposure is reduced, yet deliberate engagement with nature can help restore some of what has been lost. The hidden organ, though internal, is open to the world, shaped continuously by encounters with the external environment. Restoring balance also involves recovering from disruption caused by antibiotics or illness. After antibiotics, the microbiome may take weeks or months to regain its former diversity, and in some cases, certain species may never fully return. Supporting recovery during this time is essential. Nutrient-rich, fiber-heavy diets, fermented foods, and reduced stress can encourage regrowth. In severe cases of disruption, medical interventions such as microbial transplantation may be considered, transferring microbial communities from healthy donors to restore balance. These approaches, while more direct, reflect the same principle. Health emerges not from eradication but from the careful rebuilding of ecosystems. Stress management is another subtle yet important factor. Chronic stress alters hormone levels and immune function, which in turn affect microbial balance. The gut microbiome, sensitive to signals from the nervous system, may shift under prolonged stress, reducing resilience. Practices that calm the mind, such as meditation, gentle exercise, or restful sleep, create conditions that support microbial harmony. The relationship flows both ways. Balanced microbes support emotional stability, and emotional stability supports microbial balance. Restoring harmony, then, is not only physical but also psychological, a cycle of mutual reinforcement between mind and microbes. The concept of restoring balance invites reflection on the nature of health itself. Health is not a fixed state but a dynamic equilibrium, constantly adjusting to new circumstances. The microbiome embodies this truth, shifting with diet, environment, and life stage, yet striving always toward balance. When disrupted, it seeks to recover, and when supported, it often succeeds. Restoration is less about control and more about cooperation, less about intervention and more about encouragement. It is a process of aligning human habits with microbial needs, recognizing that their survival is intertwined with ours. In this sense, restoring balance is both ancient and modern. Ancient because traditional practices, diverse diets, fermentation, contact with soil and animals have long supported microbial health. Modern because science now illuminates why these practices matter, revealing the biochemical dialogues that underlie them. Together, they remind us that the hidden organ thrives when life is lived in connection with variety, with nature, and with rhythms that nourish rather than deplete. To picture microbial restoration is to imagine a forest after disturbance. At first, the landscape may appear barren, stripped of its richness, but slowly, seeds sprout, saplings grow, and species return. Diversity rebuilds, and with it, resilience. The forest does not become identical to what it once was, but it regains harmony, stability, and beauty. So it is with the microbiome. Restoration does not mean returning to an exact former state, but finding balance once more, adapted to new conditions yet capable of sustaining life. In the end, restoring balance is less about curing and more about cultivating. The hidden organ is not a machine to be repaired, but a community to be supported. Its resilience lies in diversity, its strength in harmony. To restore it is to provide the conditions in which life can flourish, quietly and unseen, within the body. And as this balance returns, so too does the sense of well-being that comes when the inner ecosystem is aligned with the needs of the host. It is a reminder that health is not only in the hands of human cells, but also in the cooperation of countless microbial partners, waiting to thrive again when given the chance. 17. The microbiome as an ecosystem. To understand the microbiome most deeply is to see it not simply as a collection of organisms, but as an ecosystem, a living system where countless species interact with one another and with their host in ways that create stability, resilience, and balance. Just as a forest is more than the sum of its trees, and a coral reef is more than its individual corals, the microbiome is more than its bacteria, fungi, viruses, and archaea. It is the web of interactions, the cycles of growth and decline, the exchange of signals and resources that make it function as a coherent whole. The microbiome as an ecosystem is both vast and intimate, stretching across every corner of the human body, while remaining within the boundaries of skin and tissue. It is at once part of the self and part of the larger world, a hidden wilderness that lives within us. In every ecosystem, diversity is a measure of strength, and the microbiome is no exception. A rich variety of species creates resilience, ensuring that no single disturbance can unravel the whole. If one species declines, others may take its place, carrying out similar functions and preserving stability. This redundancy is a hallmark of ecosystems, a safeguard against collapse. In the microbiome, such diversity allows digestion, immunity, and metabolism to continue even in the face of change. When diversity is lost, however, the system becomes fragile. Like a forest reduced to a single crop of trees, a simplified microbiome is more vulnerable to pests, pathogens, and imbalance. Diversity, then, is not merely a detail but the very foundation of resilience. Interactions among species are equally central. Some microbes compete fiercely, vying for nutrients and space. Others cooperate, producing compounds that neighbors use, or forming layered biofilms where species support one another's survival. These interactions mirror the relationships seen in natural ecosystems, where competition and cooperation intertwine. In the gut, for instance, one species may break down complex fibers into smaller molecules, which another species then ferments into short-chain fatty acids that nourish the host. Without the first species, the second could not thrive. Without the second, the host would not receive vital energy. These chains of interdependence form the fabric of the microbial ecosystem, woven tightly with the needs of the human body itself. The microbiome also reflects ecological principles of succession and adaptation. Just as ecosystems change over time, with pioneer species preparing the ground for later arrivals, so too does the microbiome evolve across life stages. In infancy, simple communities dominate, gradually giving way to greater complexity as diet and environment expand. Adolescence brings shifts shaped by hormones, and adulthood settles into a relative stability, though still open to influence. In old age, the microbiome often loses diversity, echoing the way ecosystems can thin as resources decline. These patterns reveal that the microbiome is dynamic, never static, adjusting to new conditions in ways that echo the rhythms of the natural world. Disturbances play a role in both natural ecosystems and the microbiome. Forests face storms, fires, and droughts. The microbiome faces antibiotics, illness, and stress. In both cases, resilience depends on diversity and balance. A forest with many species can regrow after fire. A microbiome with many species can recover after disruption. But repeated or severe disturbances can push systems beyond their ability to recover, leading to lasting changes. In the microbiome, this may manifest as dysbiosis, a state where harmful species dominate or beneficial ones decline, contributing to disease. The lesson is the same for forest and microbiome alike. Balance must be protected, and diversity preserved, for resilience to endure. Ecosystems are also shaped by flows of energy and matter, and the microbiome is no different. Nutrients from food provide energy not only for human cells, but for microbial communities. Waste products from one species become resources for another, creating cycles of exchange that minimize loss. In the gut, these cycles transform plant fibers into usable energy, recycle bile acids, and synthesize vitamins. The efficiency of these exchanges reflects the efficiency of the ecosystem itself. Just as in a thriving forest, nothing is truly wasted. Everything is transformed, passed along the chain of life until it returns to the host or the environment in altered form. Communication is another hallmark of ecosystems, and in the microbiome, it is constant. Microbes release chemical signals that influence one another, coordinating growth, movement, and behavior. Some signals extend beyond the microbial community to the human host, influencing immune responses, metabolism, and even mood. This chemical conversation is intricate, with countless voices speaking at once, yet it results in coherence. The microbiome communicates not only within itself but with the body as a whole, forming part of the dialogue that sustains life. To see it as an ecosystem is to appreciate not only the richness of species but also the richness of communication that binds them together. Ecosystems are open systems, shaped by their connections with the broader environment. The microbiome reflects this truth in its constant interaction with the outside world. Every meal introduces new molecules and sometimes new microbes. Every breath carries particles that touch the lung microbiome. Every touch of soil, pet, or person adds to the microbial pool. The microbiome is not closed off but porous, constantly exchanging with its surroundings. It is a reminder that human beings are not isolated entities but nodes within larger ecological networks, connected by flows of life too small to see yet powerful enough to shape health and survival. The idea of the microbiome as an ecosystem also shifts how we think about health and disease. Instead of seeing illness as the invasion of enemies, one can see it as the imbalance of a system. Disease arises when the ecosystem falters, when cooperation is lost, when diversity thins. Restoration, then, is not simply about killing pathogens but about cultivating balance, supporting the return of harmony. This perspective encourages gentleness, seeing health not as a battle to be won, but as a balance to be maintained, a living relationship between host and microbes that thrives in cooperation. In reflecting on the microbiome as an ecosystem, one can draw parallels to the wider natural world. Just as forests, oceans, and grasslands sustain the planet, so too does the microbial ecosystem sustain the body. Just as humans depend on biodiversity for clean air, fertile soil, and stable climate, so too do we depend on microbial diversity for digestion, immunity, and resilience. The scale may be smaller, but the principles are the same. Life flourishes in networks, balance sustains health, and diversity guards against collapse. To honor the microbiome is to honor the ecological truth that survival depends not on isolation but on interdependence. Ultimately, the microbiome as an ecosystem invites a sense of wonder. Within every person lies a hidden wilderness, rich with species, alive with interactions, and sustained by cycles of exchange. It is a wilderness as real as any forest or reef, yet it exists within, shaping life from the inside out. To imagine this inner world is to picture not chaos but harmony, a balanced system that has evolved alongside humanity for millions of years. It is a reminder that the body is not a solitary being but an ecosystem in its own right, a living partnership of human and microbial life. In this hidden ecosystem lies the quiet strength of resilience, the foundation of health, and the echo of the broader natural world, written small within the body itself. 18. Future directions in research. The study of the microbiome is still in its early chapters, though its presence in human life has been constant since the beginning of our species. For centuries, microbes were known only as invisible agents of disease, feared and fought against with sterilization, sanitation, and eventually antibiotics. It is only in recent decades that a fuller picture has begun to emerge. Microbes are not merely invaders but companions, partners. This realization has opened vast fields of research, and the future promises discoveries that may reshape medicine, nutrition, psychology, and even our understanding of what it means to be human. To imagine future directions in microbiome research is to look toward a horizon where the hidden organ becomes not only better understood but also gently guided in ways that sustain health and prevent disease. One of the most promising directions is personalized medicine based on the microbiome. Just as no two fingerprints are alike, no two microbiomes are the same. Each person carries a unique microbial community shaped by genetics, diet, environment, and history. Understanding these individual differences may allow treatments tailored to the specific microbial composition of each patient. Instead of one-size-fits-all therapies, medicine could one day adjust diets, drugs, or microbial supplements according to the needs of a person's microbiome. Already, researchers are exploring how microbial profiles predict responses to medications such as chemotherapy or immunotherapy, hinting at a future where microbiome-guided treatments become standard practice. Another frontier lies in the development of microbiome-based therapies. Probiotics, long marketed as beneficial microbes, represent only the beginning. Future therapies may involve carefully designed microbial communities introduced into the gut to restore balance or treat disease. Unlike simple probiotic capsules, which often contain only a few species, these advanced therapies might include dozens or hundreds of organisms working together, replicating the diversity and complexity of a healthy microbiome. Such treatments could be targeted for conditions ranging from inflammatory bowel disease to metabolic disorders, offering ways to heal by rebuilding ecosystems rather than suppressing symptoms. Fecal microbiota transplantation, though still a relatively new practice, has already shown success in treating recurrent infections. The future may refine this approach, replacing crude transfers with purified microbial consortia or engineered organisms designed for safety and effectiveness. Scientists are beginning to identify specific strains and combinations that exert particular effects, moving toward precision therapies that restore balance with intention rather than chance. These efforts highlight a shift in medicine, from fighting microbes as enemies to cultivating them as allies. Diet and the microbiome remain a fertile area of research. While the influence of fiber and fermented foods is already recognized, future studies may reveal deeper details about how specific foods nurture or hinder microbial communities. Personalized nutrition, guided by microbiome analysis, may become a practical tool for health. A diet that supports one person's microbes may differ from what benefits another, reflecting individuality at the microbial level. Research may also uncover new prebiotics, molecules that selectively feed beneficial species, opening doors to dietary interventions that subtly guide the microbiome toward resilience. The gut-brain axis is another frontier that promises to expand. Early studies linking the microbiome to mood, anxiety, and cognition are now giving rise to the field of psychobiotics, microbial interventions designed to influence mental health. Future research may clarify how specific microbes or metabolites affect neurotransmitters, neural circuits, and emotional states. Such insights could lead to new therapies for depression, anxiety, or neurodegenerative conditions, treating the mind through the microbiome. While these ideas remain in development, they reflect a broader recognition that the hidden organ influences not only the body but also the mind, shaping human experience at levels once thought unreachable by microbes. Advances in technology will accelerate these discoveries. Sequencing methods already allow scientists to map microbial DNA with remarkable precision, but future tools may capture not only which microbes are present, but also what they are doing in real time. Metabolomics, the study of small molecules produced by microbes, is expanding our view of microbial chemistry. Single-cell techniques may reveal interactions at the level of individual organisms. Computational models, integrating data from thousands of studies, may simulate microbial ecosystems, predicting how changes in diet, drugs, or environment affect balance. Together, these tools promise to transform microbiome research from descriptive to predictive, allowing not just observation but guidance. The exploration of microbial niches beyond the gut will also deepen. The skin, lungs, reproductive tract, and even the eyes each contain communities with unique roles. Future research may uncover how these niches interact with one another and with the body as a whole. The concept of a single microbiome may give way to recognition of a network of interconnected ecosystems, each influencing the others through shared signals. This perspective could reshape treatments for conditions ranging from skin disorders to respiratory diseases, highlighting the systemic nature of microbial life. Public health may also be transformed by microbiome research. Understanding how environment shapes microbial diversity could inform policies that encourage contact with nature, support diverse diets, and reduce unnecessary antibiotic use. The rise of chronic conditions linked to dysbiosis may be addressed not only with medical therapies but also with societal shifts that restore connections to microbial richness. The microbiome thus bridges biology and culture, revealing that health depends not only on personal choices but also on the environments and practices of communities. Ethical and philosophical questions will accompany these advances. As we learn to guide and manipulate the microbiome, what responsibilities arise? How do we balance the benefits of intervention with the risks of altering ecosystems that have evolved for millennia? The microbiome challenges simple ideas of self, showing that human beings are not solitary individuals but composites of human and microbial life. Future research will not only advance medicine but also deepen our understanding of identity, interdependence, and what it means to be alive. In imagining the future of microbiome research, one is struck by both possibility and humility. The hidden organ is vast and intricate, and though knowledge is growing, much remains unknown. Each discovery reveals new layers of complexity, new connections between microbes and human health. The promise is immense. Therapies that restore balance, diets that support resilience, and insights that connect body, mind, and environment. Yet the lesson is also clear. Health depends not on domination, but on cooperation, not on eradication, but on harmony. The microbiome, as an ecosystem within, reflects the larger truth of ecosystems beyond, that life flourishes when balance is honored, diversity preserved, and interdependence embraced. The future of microbiome research will likely bring medicine closer to nature, reminding us that healing often comes not from force, but from nurture. It will reveal that within each body lies a hidden universe, waiting to be understood, respected, and supported. And in that understanding, the path forward will not only improve health, but also illuminate the deep ties between humanity and the smallest forms of life, ties that have sustained us for countless generations and will continue to do so for as long as life endures. 19. A hidden companion. To think of the microbiome is to think of companionship, of a presence that has been with us since before birth and will remain with us until the end of life. Unlike the companions we can see and touch, the microbiome is hidden, invisible to the naked eye, yet no less constant. It is not one organism but trillions, not a single voice but a chorus, not a fleeting guest but a lifelong resident. To call it a hidden companion is to acknowledge its quiet intimacy, its tireless work in the background of every moment, and its role as a partner in nearly every aspect of health. The microbiome is not something we carry as baggage. It is something we live with as a collaborator, a companion in the truest sense of the word. This companionship begins at the moment of entry into the world. From the first breath, the first touch, and the first feeding, the newborn is seeded with microbes that will grow into communities. These early companions are not chosen but inherited, drawn from mother, environment, and circumstance. They become part of the infant's inner life, shaping immunity, digestion, and even emotional development. In this way, the microbiome is not simply an addition to human existence but a part of its foundation. From the beginning, we are not alone. We are joined by companions who shape us as we grow. As life continues, this companionship deepens. The microbiome adapts to diet, climate, stress, and environment, reflecting the rhythms of daily life. Each meal nourishes not only the body, but also the microbial residents. Each journey outdoors introduces new species. Each touch of soil, pet, or loved one adds to the shared microbial pool. In this way, the microbiome becomes a mirror of experience, carrying traces of places visited, foods eaten, and lives intertwined. It is an intimate record written in living form, a companion that remembers where we have been and adapts to where we are. What makes this companionship remarkable is its constancy. Unlike many relationships, it does not pause or rest. While we sleep, microbes continue their work, breaking down fibers, producing vitamins, generating signals that support immunity and mood. While we go about our days, they shield the skin, balance the lungs, and patrol the mouth. They are always there, unseen, yet active, living their lives in ways that support our own. Their constancy is humbling, reminding us that health is not something we maintain alone, but something built in partnership with companions too small to perceive. The companionship of the microbiome is also profoundly cooperative. Microbes gain shelter, warmth, and nourishment from the human body, while the body gains protection, nutrients, and regulation from the microbes. This mutualism is not forced but natural, the result of millions of years of coevolution. Humans and microbes have grown together, shaping each other's survival until the two became inseparable. To lose the microbiome would be to lose essential functions of digestion, immunity, and metabolism. To lose the host would be to lose the shelter in which the microbes thrive. Together, they form a partnership as ancient as humanity itself. This companion is also deeply personal. No two people share exactly the same microbiome, even though there are broad similarities across populations. Each person's microbiome reflects individual choices, environments, and histories. In this way, the hidden companion is both universal and unique. Universal because all humans carry one. Unique because each is singularly tailored. It is an invisible signature, one that sets each individual apart. Families share similarities in their microbiomes, and close relationships often lead to overlapping communities. But no two are identical. Each person lives with a companion that is uniquely theirs, shaped by their life and shaping their life in return. At times, the companionship is strained. Disruptions such as illness, antibiotics, or stress can disturb the microbiome, reducing diversity and shifting balance. In these moments, the hidden companion may falter, allowing discomfort, vulnerability, or disease to emerge. Yet even here, the companionship is evident, for the microbiome strives to recover, regrowing its communities and restoring harmony. This resilience is a testament to the strength of the bond, to the way human and microbial life are interwoven. Just as friendships endure through challenges, so too does this companionship, adapting and healing when given the chance. What is striking about this hidden companion is how much it shapes aspects of life we once thought unrelated to microbes. It influences appetite and energy, immunity and inflammation, mood and cognition. It whispers to the brain through chemical signals, guides the immune system through antigens, and supports metabolism through fermentation. These influences are often subtle, but they accumulate into profound effects. The hidden companion is not loud, not dramatic, but its quiet work underlies every heartbeat, every breath, every thought. It is present in ways that escape awareness yet define experience. To view the microbiome as a hidden companion is also to see it as a reminder of interdependence. Modern culture often emphasizes independence, individuality, and self-sufficiency. Yet the microbiome teaches a different truth, that life is sustained by cooperation, by networks of beings working together. The body is not a solitary entity, but a host of many lives intertwined. Health is not the achievement of the self alone, but the result of companionship with the microbial world. This perspective invites humility, reminding us that survival depends not only on human strength, but also on the quiet presence of countless unseen partners. The companionship of the microbiome is lifelong, but it also evolves with time. In infancy, it is fragile, establishing itself and shaping early development. In youth and adulthood, it stabilizes, supporting resilience through years of growth, stress, and change. In old age, it shifts again, sometimes losing diversity, sometimes faltering, reflecting the aging body. At each stage, the companion is present, adapting to circumstances, reflecting the journey of life itself. It grows with us, changes with us, and remains until the end, when the cycle of life returns microbes to the earth, where they join broader ecosystems once more. In reflecting on this hidden companion, one feels both intimacy and awe. Intimacy because the microbiome is part of the self, living within and upon us, shaping daily life in unseen ways. Awe because it is vast, complex, and ancient, a partner that connects each human to the web of life that spans the planet. To know that trillions of microbes live within, working in harmony with the body, is to realize that companionship is not limited to what we can see. It extends into the microscopic world, where bonds of cooperation sustain life quietly, faithfully, and without end. Thus, the microbiome as a hidden companion is more than a scientific concept. It is a reminder of the nature of life itself. We are never alone, even when we seem most solitary. We live with companions that shape us from the inside out, companions that link us to each other and to the broader natural world. They are hidden, yes, but their companionship is real, constant, and essential. To honor this hidden companion is to honor the truth of interdependence, the quiet beauty of cooperation, and the unseen partnerships that sustain the miracle of life. 20. Calm closing. As the journey through the microbiome draws to a close, it becomes clear that this hidden organ is more than a scientific curiosity. It is a living part of the self, as real and essential as the heart or lungs, though it exists in silence and invisibility. Trillions of microbes, bacteria, fungi, archaea, and viruses cooperate within us, forming communities that sustain life in ways both ordinary and profound. They digest the fibers we cannot break down, shape the immune system so it can defend without destroying, produce molecules that calm or energize the mind, and stand as guardians against invaders. In their diversity lies resilience, and in their harmony lies health. To end this reflection is not to leave them behind, but to carry forward an awareness of the quiet partnership that has always been there. The microbiome reminds us that health is not the property of human cells alone. It is shared, woven together with the survival of microbes that live in and on us. To see the body as a singular being is comforting, but to see it as an ecosystem is more accurate and perhaps more humbling. Within the human form lies a wilderness, dense and diverse, always shifting, like forests and rivers beyond the skin. This inner wilderness thrives when balance is preserved and falters when it is disturbed. The lesson it offers is simple yet profound. We flourish not in isolation but in cooperation, not by eliminating difference but by sustaining diversity. In this hidden organ lies a mirror of the natural world, the balance of species, the cycles of growth and renewal, the interplay of competition and cooperation, all echo the patterns of life beyond the body. The microbiome is not separate from nature but part of it, a continuation of the ecological web on a smaller scale. To breathe, to eat, to touch the world is to engage in exchanges with microbes, inviting them to join the communities within. In this way, each person is not only an individual but also a node in a larger network, linked by microbial life to family, environment, and planet. The recognition of this connection can be comforting, for it reveals that no one is ever truly alone. Life is shared at every level, seen and unseen. The quiet presence of the microbiome also softens the boundaries between body and mind. The gut-brain axis shows how microbial signals shape mood and perception, while immune interactions reveal how microbes calm or stir inflammation. In ways once unimaginable, the hidden organ influences thoughts, emotions, and resilience. It does so not with loud demands, but with subtle nudges, guiding the nervous system through molecules that travel in silence. To reflect on this is to realize that even the most personal experiences, hunger, calm, joy, fatigue, are shared experiences, co-created by human and microbial life. Awareness of this truth invites gentleness, a recognition that well-being is not forged alone but in partnership with companions too small to see. The microbiome also offers a vision of health that is dynamic rather than fixed. Ecosystems are never still, and neither is the microbiome. It shifts with diet, environment, age, and stress, adapting to new conditions with remarkable flexibility. Health, then, is not a static state but a balance in motion, an ongoing negotiation between species and host. To maintain this balance is not to hold still but to live in rhythm with change, allowing the system to adjust and recover. This perspective eases the pressure of perfection, reminding us that health is not the absence of disturbance but the presence of resilience, the ability to regain harmony after disruption. As modern research continues to uncover the microbiome's secrets, new possibilities open for medicine, nutrition, and mental health. Yet, even without advanced therapies, the core truth remains. The hidden organ flourishes when nourished with variety, connection, and balance. A diverse diet, time in nature, rest, and calm are not only beneficial for the body, but also for the microbial communities that sustain it. These simple, timeless practices reflect an ancient wisdom, one that humanity carried long before science revealed the details. They are reminders that caring for the self includes caring for the companions within, that the health of one depends on the health of many. To close this reflection, it may help to imagine the microbiome not as an abstract concept but as a presence. Picture a vast, quiet forest, not outside but inside, spread through the winding corridors of the gut, across the broad plains of the skin, and into the smaller niches of the mouth, lungs, and eyes. Picture each tree as a species of microbe, each leaf as a single cell, all working together in unseen harmony. This forest is alive within, growing and shifting each day, sustained by what we eat, how we live, and the world we touch. Though invisible, it is real, a companion whose roots are as deep as human existence itself. In the stillness of night, when the body rests, the microbiome continues its work. Fibers are fermented into fuels, immune cells are guided, and signals travel to the brain. This hidden activity sustains life quietly, asking for nothing in return but balance. To fall asleep with this awareness is to feel a kind of comfort, a reassurance that within, countless companions labor gently to support the body's well-being. Their presence is constant, their loyalty unwavering, their cooperation enduring. They are not invaders, not strangers, but fellow travelers, sharing the journey of life in ways too small to see, but too important to ignore. Thus, the calm closing of the story of the microbiome is not an end, but an invitation to remember. Remember that health is shared, that diversity is strength, that resilience is balance regained after disturbance. Remember that life within mirrors life beyond, and that in every breath and every heartbeat lies a partnership between human and microbial existence. The hidden organ is not apart from us, but a part of us, a quiet companion that has always been there and will remain as long as life endures. To honor it is to honor the truth of interdependence, the quiet resilience of ecosystems, and the beauty of cooperation that sustains all living things.